Thank you very much for being here. Thank you. Thank you for coming. So my name is Olga Vitek. I am director of the Barnett Institute for Chemical and Biological Analysis, and it is my great pleasure and my great honor to welcome you all today for the celebration of the Barry Karger Medal in Bioanalytical Chemistry. And the plan for today is to spend the first part of the celebration presenting some of the work that the Barnett Institute faculty fellows are doing, and hopefully it will be of interest to many of you. And then we will have a break that will allow us to continue the many conversations that we just started, and also have a poster session where some of the students and members of the Institute present their ongoing work. And after that, we will have the main event of today is the actual medal award ceremony and presentation by the awardee. So to start, and without any further ado, let me welcome Professor Hazel Sive, the Dean of College of Science, who will have some welcome remarks.
Thank you so much, Olga. What a pleasure to see you all. I'm Hazel Sive, Dean of the College of Science here at Northeastern University, and it's my pleasure to open this important Karger Medal event. I want to give my warmest congratulations to Dr. Bernhard Küster, the recipient of the Karger Medal. We're honored to have you join us at Northeastern today. Welcome to Dr. Barry Karger. It is always such a pleasure to see you. Welcome to you all, current Barnett Fellows, faculty, students, postdocs, alumni, collaborators from the industry, and friends. The Barnett Institute is one of the top research institutes at Northeastern University, founded by Dr. Karger more than 50 years ago. And as many of you know, we had a wonderful celebration of this important founding not so long ago. Under the present leadership of Dr. Olga Vitek, I'm thrilled to see the Barnett Institute move forward with energy and highest quality research that includes the cutting-edge techniques that will be explored today. Congratulations to you all. Today's symposium, with presentations, posters, and discussion, is science research at its pinnacle, focusing on proteomics and drug discovery, including integrating AI approaches. Each speaker is a leader in the field with spectacular contributions coming from across the university, including from the chemistry department, and I'd like to acknowledge the chair of chemistry and chemical biology, Professor Penny Buning, who is here today. Welcome, Penny. Each student or postdoc presenting a poster is receiving top quality training, furthering your acumen towards an excellent research career. You know, I'm wondering if we could, there's a little bit of background there. Thank you so much.
Why don't you put the microphone down a little bit?
Are you having trouble hearing me, Barry? Okay. Thank you. I have been accused of having too loud a voice, so I love it. I can be real loud. We talk about fundamental or applied and translational research. But in my view, all science research is translational. All science research becomes translational. It's simply a matter of when. For example, one of our favorite topics, mRNA, was discovered in 1868 by the Swiss researcher Friedrich Mischer, a chemist who was not looking for RNA, but stumbled across it. Over the next 100 years, millions of hours of research time and several Nobel Prizes, it became clear that among many activities, RNA carries the code that directs the production of proteins, a topic of great interest to you all in this gathering. 50 years after that, stabilized, modified RNA, in conjunction with the knowledge of the membrane that encases a cell and the chemistry of lipids that make up the membrane, could be combined into mRNA vaccines. It's a long way from the discovery of nuclein, the original name for RNA, 150 years ago. This history makes my point that every discovery in every field will someday have an applied use. And I urge you, when discussing your own scholarship, to bear that in mind and to emphasize that the distinction between discovery and application is always there. It is not one or the other. Now, in addition to celebrating your exciting research at various stages of application, today I want to explore with you another aspect on my mind and leave you with a challenge. The research that you do is incredibly sophisticated. You understand it, but few others do. You receive good funding for it, but few understand why. The why do science research question is ever present because our work is complex and hard to understand. In the new US administration, the power of research seems especially poorly understood, with federal funding agencies targeted, as you know, in a very fluid situation. Each of us must meet these challenges and protest incursion on funding through our elected officials, our professional societies, and more directly. That is important and useful. We must each stand up for science in these ways. But there's another way to stand up for science that may also help science funding. People not working in science don't understand the importance of science or science research. If there is to be broad public support for science research, we, each of us scientists, needs to explain what we do, what science does, at the most simple, clear level, to everyone, regardless of their education, their job, or their views. It is our responsibility to explain this. It is your responsibility to explain what you do and why science is important. So how can we explain the value of science? There's a scenario that can help. I call it understanding by subtraction. Let's start with this group. Let's say science research of any kind never existed. Where would this symposium be today? Well, there would be very few people to present as a start, since most of us would have succumbed to myriad illnesses long ago. Those who were here would not be in this room, since the science and technology for this building would not exist. We would not be wearing our comfortable clothes. We would be very cold without our Navian boilers. We would not have had a good cranberry muffin and coffee for breakfast, since the science behind cooking would not exist. And we could certainly not have taken the tea or an Uber here today. You get the idea. It's a great way to explain to anyone the incredibly broad, powerful reach of science. It also works well to flip it and ask someone, anyone, how science has helped their family. You'll hear stories about medicines, surgeries, travel from a distant place, stories of survival made possible by science. You might also hear about baking bread, painting your house, nail salons, and airplanes. Science did that. And once you have your neighbors, any person, any group, thinking about how science is useful and not scary, and how they would likely not be here without science, then you can explain what science research you do and why it's useful. I would charge you, when you explain what you do, not to use any jargon or science terms at all. It's quite possible. A couple of years ago, I gave a TED talk here at Northeastern in my field of developmental biology. I talked about how a human was built, and I used only 3 scientific terms in the talk: cell, gene, and DNA. People loved it because it was easy to understand. You needed no training. When we explain this way, science research loses its elite sheen, loses the sense that research is only for the few privileged or talented enough. When we explain that science is useful and what we are researching in simple terms, science can become accessible. This understanding helps empower government officials locally and nationally. It helps every voter understand why research is really important. While working in highly sophisticated fields, we need to understand that explaining the point to everyone who wonders is up to each one of us. I'll tell you a story. My brother is really intelligent. He's well educated, but not in science. We have conversations all the time about science. And he still asks me questions like, are proteins alive? This is true, okay? We have to speak so everyone can understand. I charge each of you here to speak with whomever will listen, explain science research, your research, bring people in. It's always necessary, not just in this administration. We are always fighting for funding, although it seems a bit more dire at the present time. Let us stand up for science together, broadly and accessibly. As the swirl of surprising actions against the NIH, NSF, and other agencies continues, I am, however, optimistic that funded science research will go forward well enough. We are encouraged that review panels are happening and thrilled that awards or intents to be awards are being communicated to our junior and senior faculty in the Northeastern University College of Science. Let's hang in there and take every opportunity to explain why what we do is important. I wish you a wonderful symposium. Congratulations again to Dr. Küster. Congratulations to you all. Thank you.
Thank you, Dean Sive, very much. So now let's move on to the next point in our program. I would like to welcome Professor Nikolai Slavov from the Department of Bioengineering at Northeastern University, who will be telling us about his work on single-cell proteomics, among other things.
Thank you so much for the invitation and the privilege to present at this symposium. I considered how can I recognize best the contributions of Bernhard Küster to our community and to reflect what he is best known for. And I thought that the most salient, well-recognized, deservingly recognized contribution is that Bernhard has contributed many high-quality tools and data sets to proteomics, to biomedical research, and that's why I chose to share with you a project, the first project that I'll share with you from my group, that has made very active use of data sets and tools that Bernhard's group has developed. And then in the second portion, I'm going to tell you about some recent single-cell work that indeed allows to not only do measurements in single cells, but use those to learn things about biology, about biological mechanisms of regulation that otherwise we could not have learned. So we are going to talk about proteins and proteomes, and it's important to start by acknowledging the tremendous complexity of proteomes. It is not just the genes encoding them. It is multiple processes such as alternative splicing, post-translational modifications, protein cleavage, coordination, protein-protein interactions that results in very complex protein mechanisms that ultimately make life possible. And when we think of what governs protein abundance, we usually think of the left. We think of the synthesis. But there is another component that is very important, which is the degradation. There are two sides. There is the rate of protein synthesis, and there is the rate of protein degradation. And I want to highlight here a recent result from Andrew Leduc, a postdoctoral fellow in my group, where we wanted to examine what are the relative contributions of protein synthesis and protein degradation to shaping the proteomes of mammalian cells. And what we found is that perhaps intuitively, when cells proliferate very fast, such as cells in culture, the primary mechanism for removing proteins is dilution. But when we consider cells that are non-proliferative and non-growing, such as the majority of the cells in our bodies, such as the neurons in my brain that make my speech possible, then we see that degradation, shown here, actually explains a very, very large fraction of the variance of protein abundance, and we could follow this systematically and identify what are the factors explaining how much of protein variation is determined by protein degradation. And we see that regulated changes in the rates of protein degradation are a major, major factor that has not received much attention. Now, how do we know the sequences of the proteins that make our bodies, the other side of the production equation? We mostly infer those. We've never been able to directly determine sequences. Perhaps Barry Karger remembers a time when there was a hope that we are first going to determine protein sequences before we knew how to sequence a genome, but then we sequenced the genome and made those indirect inferences, and those indirect inferences were greatly facilitated by the genetic code that was uncovered by Marshall Nierenberg in the intramural wing of NIH from in vitro synthesized RNAs that were translated by E. coli cell extracts. And E. coli translational machinery is very different from the one in our cells. E. coli has over 10 times fewer tRNAs, much simpler, much smaller ribosomes, fewer translation factors, and so on. But nonetheless, it's a remarkable, remarkable fact that a genetic code inferred from artificial synthetic RNAs and E. coli extracts has been so useful to be able to predict protein sequences across a variety of different organisms. But nonetheless, we know that the genetic code doesn't explain all protein sequences present in mammalian cells and in vertebrate cells in other organisms. And the type of measurements that people have done for a long time is either using 2D gels with radioactive isotopes long ago, or more recently using reporter genes based on fluorescent constructs whose when the incorporation of alternate amino acid is detected as change in fluorescence, and the results from these artificial constructs suggest that there is a substantial rate of alternative RNA, of alternative amino acid incorporation. And we wanted to examine to what extent those deviations also happen with endogenously encoded human proteins. And to do that, we use this very simple expectation that is well understood and familiar to all colleagues in the audience, that if we have a change in the amino acid sequence in a peptide, we are going to alter the masses of the fragments, the mass of the precursor, the retention time, the separation of the peptide, and we can use this evidence to identify hypotheses for that substitutions. And we did that by using data from over 1,000 human proteomes and transcriptomes, including data from the lab of Bernhard Küster. And we started by selecting samples for which we had very deep sequencing because we wanted to be able to characterize what is a transcriptome, what are the messenger RNAs templating proteins, and then determine peptides that have deviations from the expected sequences. And of course, we identified a large number of modified peptides, about 9 million of those, and many of them could correspond to either artifacts happening in sample preparation, to known post-translational modifications, and also to many amino acid substitutions. There were about 60,000 of those. And that is, of course, something that requires very, very rigorous evaluation and validation because it's surprising and we want to be able to have confidence in any inferences that we make. So let's first look what are these proteins that we find. And we looked both at the abundance of the protein whose sequence is predicted by the genetic code and the abundance of the protein whose sequence is not predicted by the genetic code. And quite surprisingly, we saw that for many proteins shown at the bottom, transcription factors and signaling proteins and receptors, the abundance of the substituted version that wasn't predicted by the genetic code was rather high, in many cases more abundant than the sequence that we know about, and we were very, very surprised about this. So we did a number of different evaluations that are detailed in a research article. I'll just highlight a few, and I'll highlight specifically a way in which a tool developed by the Küster lab was helpful for us. But first, let's look at the raw data. If we have a surprising observation, it really helps to look at the bits and see, can our human brain see that? And here is one example where we can detect 86 RNA reads that are perfectly complementary to the genome and to the reference databases, and they correspond to a peptide sequence, which is shown on the left. And for that, in the mass spectra, we see that there is very strong support for that peptide. And we also see on the right that there is another peptide for which there is an amino acid substitution. Instead of asparagine, we have alanine, and for that we also have a beautiful spectrum with strong support for its existence, and this is the more abundant version of the protein. It's actually 15.8, about 16-fold more abundant. So based on looking at those examples, this looked credible to us, but can we do this more systematically? And here we used a deep learning tool from Küster's group, PROSIT, that is able to predict the intensities of peptide fragments. When we fragment them inside of mass spec instruments, we can use that information to either increase or decrease our confidence in identified peptides, as has been demonstrated many times. Similarly, we can predict retention time and use it for that. And we see that when we rescore our confidence, it goes up by about 10-fold in most cases. So most of those newly identified peptides with sequences not present in any prior databases are actually even more confidently identified when rescored. And another way, again afforded by experiments done by Küster and colleagues, to look for that is to see, are these different sequences identified if we use different enzymes to digest the proteins? We are going to generate different peptides with different sequences, but we should still be able to identify the same amino acid sequence substitution with the same abundance, and by and large, we see that is the case, that using different proteases reinforces the existence of these different ways of decoding RNAs. Now, I'll tell you just a tiny bit about mechanisms contributing to that before I jump to discuss other topics. The first and obvious one is, what is the contribution of incorporating alternate amino acids to translation deviations to the abundance of these proteins? And here we see something quite expected and reassuring, that when the mismatches between the codons and anticodons involve only a single nucleotide, the abundance of incorporating different amino acids is 50-fold higher. And this strongly suggests that the rate of synthesis has a contribution to the abundance of these newly discovered proteins. We can also see that the relative frequency of codons at which those substitutions are observed is a strong predictor for the abundance, for the ratio between the substituted and unsubstituted version of the protein, and that is again another suggestion for the involvement of synthesis and translation. We also see a strong correlation with RNA modifications, such as pseudouridination. If a uridine is converted to a pseudouridine, there is a higher probability of seeing incorporation of an alternate amino acid, and there is a lot deeper analysis described, but I'll just quickly switch to the other side, which is, I just told you, is not less important. It is the side of protein degradation. Is it possible that in some cases those substitutions actually stabilize proteins? I think our default model is that whenever there is a deviation in the sequence of the protein that is synthesized is likely misfolded, unstable, quickly degraded. And that's probably true. But what if in some cases the alternative amino acid sequences for alternative proteins are more stable? And this is something that with metabolic pulse of amino acids labeled to its stable isotopes, we can directly measure this. And here we see that in such experiments, the highly abundant, alternatively translated proteins show much lower rate of degradation, much higher stability. Thank you. Much higher stability, and this really opens an interesting question about the possibility of such proteins accumulating in our bodies with aging, and there is some preliminary evidence for that happening with this shown peptide here on the left in the case of Alzheimer's disease patients. But very early days of identifying those forms of proteins that have been harder to predict. And we can also, my team developed a whole model to predict the frequency and the abundance of such proteins, products of alternate translation, and we are able to predict that with good accuracy. And we are also able to identify many amino acid motifs and signaling pathways that are associated with it. In some cases, we see that these alternative sequences are strongly associated with diseases, as shown here in panel A, where a particular peptide is much more abundant in tumors compared to surrounding normal tissues for 3 different types of cancer. And there are also very interesting distributions, spatial distributions of these substitutions along the linear and the 3-dimensional sequence of proteins. But I'll stop here on the side of this project because I would like to share some other work from my lab, and just to acknowledge that much of this work that I presented here relied on data sets and tools from the Küster group, and thank you for that, and thank you from the whole community. I know that we are not the only ones using those tools and data set. There are many, many people who are benefiting tremendously from the high quality data sets that you're known for. And with that, I'll switch and talk a bit more for single cell proteomics, more established work, giving you a very quick overview of the technology and then some highlights of what's about to come. And of course, biological results. Now, at the moment, we are able to prepare samples for thousands of single cells per day, in large part thanks to Andrew Leduc developing the methods for that. We are able to acquire data for dozens to thousands of single cells, depending on the methods that we use and whether they're multiplexed or not. And usually per single cell, we can count order millions of protein molecules that map to order thousands of gene products, proteins, and proteoforms. So as a big picture, for the sample preparation has clearly been an important area of development. How do we deliver efficiently proteins from single cells to instruments while keeping contaminants out? We've developed several generations of methods to do that. The latest generation that is very versatile and we use extensively is based on these tiny droplets on the surface of glass slides. They're microscopic glass slides for imaging, where each droplet has only a few picoliters of buffer and serves as the reaction vessel where a single cell is isolated, lysed, proteins digested, labeled if need be, and then pulled together and transferred to an autosampler of a mass spec instrument. And thanks to work done by Leduc, this has become highly robust, automated, and able not only to produce lots of samples in terms of parallelizing, increasing throughput, but also very importantly, achieve high quality data by keeping contaminants out and keeping peptides in. And one of the really important early realization for us was that because of the awesome technology of mass spectrometry instruments, we have the raw sensitivity to detect many thousands of peptide-like features from individual cells. And the main challenge is determining confidently their sequences. And my laboratory has developed 2 solutions to determining peptide sequences more efficiently. The first one is what I do in my personal life. I have a very long to-do list. I can never do it all because I don't have the time, so I prioritize. So we do exactly the same thing with the mass spec instrument. We use real-time instrument control based on measuring retention time and M/Z to determine which are the peptides coming from proteins of interest with post-translational modifications, with the relevant properties that are most important to the project, to spend our limited time. And this also allows us to increase depth of coverage, data completeness, overall number of identified proteins by about a factor of 2. We are able, even when using isobaric mass tags, which generally results in low data completeness, we are able to bring data completeness well over 90% for single cells. It's something that really exemplifies the power of strategy and logic, using exactly the same equipment as before, just changing the logic of how we operate the instrument. We can realize very significant gains in data quality and completeness. The other approach is something that I'm much less successful at implementing in my personal life, which is multitasking, doing lots of things in parallel, and this has its perils, but I'll tell you a bit more about it. So the idea is that we would like to be able to label our peptides from different samples so that we can mix them together and analyze many samples at the same time. And at the same time, we want to be fragmenting multiple peptides, which generates complex spectra, harder to interpret data, but that's a challenge we wanted to take and see if we can rigorously do that while still producing high quality data. And here I'm showing some raw data to give you a sense of how they look like. On the top, you can see the elution of a peptide ion from PDAC cells. And on the bottom, the same peptide sequence from a monocyte cell. So we are able to acquire 3 data points, giving us estimates for the relative abundance of that peptide. Similarly, we need to fragment the peptides to determine their sequence. So from the fragments, we obtain another 3 data points. And from the level of consistency between these estimates, we can derive our confidence, whether that's low or high in the measurements. And if we zoom out of this specific example, we see that across many proteins, we are still able to achieve very good quantitative accuracy and precision at the relative level, and also an excellent dynamic range of about 1,000-fold variation at relative level. Now, those developments were noticed by, by Eric Schmidt and Ken Griffin, who approached us, and Convergent Research. They approached us with interest of exploring whether investing more resources in this direction can allow us to accelerate the pace of development, and this resulted in starting a new non-profit research institute, Parallel Squared. And there we have multiple objectives on the side of biomedical research and also technology development, and I'll give you a snippet of some technological progress that we've made. So we want to engineer small molecules to use to label peptides so that we can improve sequence identification, we can improve localization of post-translational modifications, we want to improve accuracy, depth of coverage, sensitivity, all of these things. Can we do it? It's actually quite challenging, as it turns out, but nonetheless we tried, and I'll show you some preliminary results of that. So here is a particular peptide that is either analyzed without a label, what we normally call label-free on the top, or it's labeled with a molecule that we synthesized, we call it tag 6, and we can look at the peptide fragments when we fragment this peptide in the instrument. And you can see that label-free in this case actually gives us fairly good coverage, but the labeled peptide has substantially more fragments being detected. All of the B-ion series is present in the tagged peptide, much less represented in the label-free peptide. And what I'm showing here is not just a cherry-picked example, but when you look more broadly across many experiments and many peptide sequences, and we compare those scores, we see that the labeled peptides generally have many more fragments, more intense fragments, and they give us much higher confidence in sequence identification, or also de novo sequencing. So if we go back to my first talk about being able to identify what the human proteome really looks like, it's really useful to have very dense coverage of those peptide bonds so that we have a chance to de novo identify those sequences, or if matching them, we have a chance to match them with high confidence and not misinterpret sequences. And we are going to have a symposium here at Northeastern about a month from now from PTI to share much more technology, and very much intend to make those tags available to everyone. They're available not only for multiplexing increasing throughput, but as I'm showing here, also for increasing the depth of coverage and data quality for de novo sequence, localization of post-translational modifications, and so on. So in the final few minutes, I'll run very quickly through an example of using single-cell protein measurements to learn new biology. And the goal is to use protein covariation across single cells to do those inferences. We focused on studying protein transport from the cytoplasm to the nucleus. We asked the question, how differences in the proteome state of macrophages influence their response to LPS in the very first few minutes of the stimulation as proteins are transported from the cytoplasm to the nucleus. And to do that, we used Plex DIA with a lot of benchmarking here shown to demonstrate that we are able to quantify a good number of proteins in single nuclei, over 1,000, while maintaining very high accuracy and precision. And based on these data, simply correlating protein abundances in the initial state of the macrophages to nuclear transport, we are able to identify a large number of proteins whose abundance is predictive of the response of cells. And these proteins include, perhaps not surprisingly, members of the nuclear pore subunits. And this directly indicates that nuclear pores are kinetically limiting the rate of transport. And furthermore, the data are suggesting that different parts of the nuclear pore have different contributions to that transport, where the inner ring subunits are more strongly correlated, and that difference is also statistically significant and associated with differences in the degradation rate of these proteins. So that's an example where single-cell measurements can suggest plausible hypotheses about complex subunit interactions and how complexes work at a much finer molecular level. And we are also able to identify a large number of additional proteins that are associated with transport as putative regulators that Jason Dirks, a postdoctoral fellow at the time in my group, followed up by doing siRNA perturbations and was able to demonstrate that the vast majority of those inferences from protein-protein correlation were validated by the targeted perturbation using siRNAs. Inspired by Bernhard and other colleagues, we are very passionate for sharing data. We make data portals where we share metadata, raw data, process data. We've also worked with leaders in the community to establish best practices for single-cell proteomics to facilitate wider adoption. And these are the people in my group at Northeastern who have made the work that I presented not only possible, but also very, very enjoyable and fun. And of course, the funding agencies and sources that have enabled the research. I'll stop here and I'll be happy to take questions if we have time.
Thank you so much, Nikolai. We do have time for a few questions if anybody would like to ask Nikolai. And we have a microphone in the back, so whoever would like to.
And I should say that I really, really, really love questions. As some of you know, we organize a single-cell proteomics conference here at Northeastern, and we spend at least half of the meeting time in questions and discussions.
So we have a question in the back.
Thanks for the talk. Could you comment on possibility of using this platform to apply on the metabolomics, for example, like single cell?
Absolutely. Single-cell metabolomics can be performed. There are many colleagues who work in that direction. It has some shared challenges on the side of sample preparation. For example, the sample preparation that we've developed in many ways can be easily adopted for metabolomics. There are additional challenges with metabolomics to identify many of the metabolites because fragmentation is much less predictable. There are challenges in identification, but there are certainly many metabolites that can be quantified well. And in my mind, one of the major challenges with single-cell metabolomics is preventing changes in metabolites such as oxidation before they enter the mass spec instrument.
Thanks.
Any other questions?
Thank you very much for the very nice presentation. I actually have a question about the first half of your presentation. I also read the paper paper when you published the preprint. It's very interesting, very great work. So my question is, did you check this amino acid changes per direction? Because there are some work that was done in the 70s where they looked at this codon changes, and they found out that in the first few amino acids, first 20, it's actually quite important because of the ribosomal machinery and the availability of tRNAs. It changes the speed of translation, thus affects the stability of the protein. Sometimes the machinery can fall off. So I was wondering if you check the codon bias in these peptides.
So we certainly see that codon bias affects the probability of having amino acid incorporation deviating from the genetic code. There is a strong correlation with that. Codon bias, the frequency of codons. Also, there's a whole dimension of tRNA modifications that is correlated to that. The substitutions tend to have distinct signatures for different organs and cell types, which is also consistent with different tRNA modifications, translation factors, differences affecting them.
One last question. Are there any changes per position? Like, does it change per position, like the changes at the beginning of a protein?
Across the protein, yeah. So there is a very slight trend of that. Yes, it becomes more nuanced because it depends a bit on how we normalize data, how we apply stringency of filtering. So there isn't a very strong bias in terms of the first part having much higher rate of alternative translation than the second half, but there are some relatively weak trends and that require more context for interpretation. There are some, yeah.
Okay, I think that's all the time we have for questions. Let's thank Nikolai.
Thank you.
So next, I would like to welcome another faculty fellow of Barnett Institute, Ben Gyori, who is associate professor in the Khoury College of Computer Sciences and in the Department of Bioengineering. And Ben will tell us about AI-assisted knowledge integration for protein biology.
Thank you. Hi, everyone. Let's see, slides. So what I will talk about now is ways in which we can use machine assistance to solve this problem of knowledge fragmentation across literature and various databases, and using such assembled knowledge to improve data analysis in a more principled way. So in both lab and increasingly clinical settings, the types of omics data, whether it's at the level of genes, transcripts, proteins, or metabolites, is increasingly collected at large scale. And the bottleneck is becoming one of interpretation, because irrespective of which type of omics data we are talking about, very often the type of end product you get from the initial analysis of such data sets is a list of genes, proteins, metabolites, and so on. But the types of questions we often want to answer from these high-throughput data sets is, like the ones on the right-hand side there, what is the mechanism behind a given increase or decrease in a readout? Which of these observations have others made? In what other contexts? Which observations can be explained by things that we already know as background knowledge? And which are things that are surprising? So what can we overall learn from this data set that is not trivially explained through background knowledge? And so my claim is that to answer questions like this, it requires integrating data with the vast amount of prior knowledge that is available, and to construct knowledge-driven explanation systems. So this is a depiction of what that background knowledge might look like, at least at the level of cellular behavior. So a lot of complexity comes from the fact that anytime we investigate cells through perturbation, those perturbations are not perfect. Drugs have multiple targets at different levels of strength. There's a lot of crosstalk between traditionally identified pathways, feedback effects, microenvironmental effects. Phenotypes of cells are not unimodal. There are multiple phenotypes that happen simultaneously and have to be kept track of. So it's quite a complex picture. So on the two sides of this, the bottlenecks become on the left-hand side, from the perspective of data, the semantic technology that's necessary to integrate data sets and make data sets interoperable and connect them to background knowledge, which itself has to be assembled from multiple different fragmented sources with as much mechanistic detail preserved as possible. So this is an overview of the kind of vision we might work towards to solve these issues from a perspective that Olga, Vitek, and I wrote last year. So on the top, you see that the three types of input are literature and knowledge bases, as well as existing data sets that you might want to leverage to interpret your own data set. And having collected some new data, like the dish on the left-hand side, you want to leverage this external knowledge to explain mechanisms that might serve as a hypothesis for follow-up experiments. So the first thing I'm going to talk about is this issue of trying to reuse existing data or trying to align data sets with external knowledge through semantic interoperability techniques. This issue has been recognized in various large efforts by funding agencies as well as community projects. Some examples here on the left-hand side, the latest one that I'm also involved in, ARPA-H, Biomedical Data Fabric Toolbox, the NIH's Illuminating the Druggable Genome, NCAD's Data Translator, some community efforts like the Understudied Proteins Initiative, which have recognized a list of issues related to data integration. And one has to do with fragmentation and incompatibility. So data and knowledge is distributed across multiple sources in a way that is not standardized. It's very difficult for authors, even if they are willing to make their data public, which is still not fully an adopted approach, but increasingly so, to make their data fair and interoperable, it requires quite a bit of effort. And so decreasing that effort through automation and machine assistance would create a major boost in those efforts. Data integration is slow and cumbersome. Whenever you try to use a data set, there's a lot of work involved in mapping that data set into the right identifier space, pre-processing it in various ways, which itself creates a bottleneck, and several other issues that basically all of these programs, one way or another, are trying to address. So this is a recognized problem in the community. So we understand these problems, but where do we start? Well, building from bottom up, the first issue is how do we even identify concepts that we talk about in the biomedical space? So proteins, diseases, cell lines, small molecules, and so on. The community is increasingly adopting the use of what we call compact universal resource identifiers. Here's an example where CABI, it tells you what the source is for an identifier, the namespace or the database or ontology, followed by a locally unique identifier within that resource that tells you the specific entity that is being referenced. So here's an example of Ulster pollen. It's a small molecule that's pretty widely used, and it has an identifier in the CABI database. People traditionally also use these universal resource identifiers, but those are actually problematic in many different ways, partly because of their own heterogeneity. So you can actually express a given entity's identifier in at least 5 or 6 different ways using URLs, so those are not standardized enough. But the question then is, okay, so this is how we want to uniquely reference things that we talk about in biology. What are all the databases that contain identifiers for things, and what are valid identifiers within those databases? It turns out that there isn't or hasn't been in the past a comprehensive resource to address this. So we built the bioregistry, which for every identifier's resource that is known, contains a meta-registry entry. For instance, there's a screenshot of the UniProt entry, which assigns identifiers to proteins, among other things. It contains metadata on the resource itself, its creators, what valid identifiers are, how to resolve those identifiers to a description page, and so on. And it does this for on the order of 2,000 such identifiers resources. And it's interesting to look at, for instance, a resource like the NCBI taxonomy, which is the primary resource for assigning identifiers to various species, that it's being referred to in different places under very different terminology. So some call it taxonomy, others call it NCBI taxon, others call it NCBI_tax ID. Even the way we refer to these databases is inconsistent, and so the bioregistry harmonizes across these, and you are able to take all of these different inconsistent usages and basically standardize them. And the two colorful plots here, what they show is that there are some existing registries similar to this, but there are many of them. None of them have comprehensive coverage, and the overlap between these registries that try to catalog these identifiers resources is highly lacking. So there's no consistent overlap between them. And so increasingly we are using various machine learning based pipelines to automatically discover resources from literature as soon as they are published that can be integrated into the bioregistry, like a new database for a certain type of interesting concept that we can integrate. And also for the automated curation of this meta registry that maps across outside sources. These are increasingly part of machine-assisted pipelines to scale up the curation effort. Okay, so now we know how to create unique identifiers and use unique identifiers for different concepts like proteins and small molecules. But given some unstandardized piece of text that might contain these, how do we assign identifiers to entities of interest? So this is often called named entity recognition or named entity normalization in computer science. The idea is that you can build on both dictionaries, like large collections of names and synonyms for things that are known, and various types of machine learning approaches to automatically look up possible matches for a given entity's name, and then perform disambiguation to determine which of many possible ambiguous usages is relevant in the given context. Because a lot of the terminology that is used, whether it's literature or databases, is ambiguous. So ER, for instance, can stand for estrogen receptor emergency room, endoplasmic reticulum, and so on. And it really depends on context which one is relevant. And so then you might imagine how, given a data set like the one here, which contains drugs, protein, phosphocyte readouts, and a set of cancer cell lines, you can actually use a tool like this to automatically look up identifiers for each of these and thereby connect them to this set of outside databases, but also to each other. So through this absolute reference that you get from identifiers, you can now take a different data set that might use different names for its cell lines, also non-standardized, but through that identifier assigned to the cell line, you might find that they are actually talking about the same thing. Our problems are not over yet. Even if you do all this, there's still the issue that there are different widely used databases that assign identifiers to things that are overlapping. Here's an example of prostaglandin E2 that is given an identifier in all of these different databases: KEBI, Campbell, PubChem, MeSH, AgeMDB, CAS, DrugBank, right? So, and different sources will give you different identifiers from different ones of these databases. So you have to have a machinery to map across these to do data and knowledge integration. And that can happen through graphs like the one that you see here. However, the mappings that are provided from some of these databases to each other are themselves incomplete, so there's a need for targeted machine-assisted curation of these missing mappings to improve data integration. And so one of the relatively newer things that we've built is a large graph database of all of the mappings that we could collect from more than 100 different sources, containing identifiers from like 445 different identifier databases. All of these mappings look like the one on the top left there. So for instance, a disease in the disease ontology mapped to the same disease in the experimental factor ontology, supported by different pieces of evidence. And compiled into a graph so that you can do reasoning across it. You can, for instance, perform indirect mapping chains where things are not directly mapped to each other. You can do that through this graph data structure. And then we created projections of this for disease identifiers, anatomical terms, protein identifiers, gene identifiers, cells, and cell lines. And then you can use these to integrate data where various different diverse identifiers are used across these types of entities. This is a kind of an interesting example here related to cell lines. So this is a single cell line that's represented in 9 different databases under different identifiers, and the edges between them are the pre-existing mappings that are provided between these databases. So you can see that it's completely ad hoc, right? So it's not that there's a source that comprehensively gives you all of these mappings across these databases. It's really ad hoc which one map to each other. So unless you integrate across them, it's hard to do this kind of large-scale data aggregation. And so ultimately, these tools will be built into AI-assisted workflows. And this is an example from our collaborator at NYU, Juliana Freire's group, who have been building LLM-based agents that can do schema mapping from some source data to a schema that is used in one of the community data portals like Proteomics Data Commons to facilitate authors' deposition of their own data sets into these databases through a combination of these semantic technologies that I talked about and a back and forth flexible natural language-based AI assistant approach. Okay, so far I talked about AI-assisted semantic interoperability, which is really the basis of data and knowledge integration. Now I'll move on to this second box here, which is about how we can collect prior knowledge from literature and databases, assemble it in a standardized form, and then generate actual models and networks from that for data analysis. So the basic concept behind this is to tap into existing pathway databases that talk about protein function, but also other types of entities like drug target databases and so on. The vast amount of literature out there, the scale of this is about 35 million entries in PubMed currently, about 5,000 new papers a day, as well as in some cases direct expert input or curation. This goes into a layer of knowledge assembly, which accounts for providing a representation for the knowledge, importantly, but also performing error correction, redundancy resolution, so the same mechanism being described in different places, the alignment of those, assessing relevance for a given use case, assessing believability, and inferring missing information. That's this layer of knowledge assembly. And from that intermediate knowledge level, you can generate different end products like executable models, mechanistic networks, which are specifically tailored to a data analysis task. This conceptual idea is implemented as software in our Indra system, the Integrated Network and Dynamical Reasoning Assembler, whose software modules are designed in a way that they really map onto this conceptual architecture with various interfaces towards text mining systems and pathway databases on the left-hand side, central knowledge assembly and knowledge representation layers in the middle, and output assemblers on the right-hand side. The types of databases that we take into account, this is just a subset of them, but you might be familiar with Pathway Commons, which includes Reactome, Senior Pathway Database, Comparative Toxicogenomics Database, and so on. So these are some of the sources that are taken as input in Indra. But more interestingly, we use a panel of different text mining systems to process literature, to extract mechanisms being mentioned in literature, like the ones that you might see from just looking at the titles of these papers, each of which is like a piece of a puzzle describing some specific part of cellular behavior. This is a classic NLP system that takes a sentence like the one you see here, ESBP2 can be phosphorylated on serine 827 by MAPK1, and turns it into an interpretation graph from which you can then conclude that there's a phosphorylation relationship being described here with a given kinase, substrate, and site. More recently, of course, these large pre-trained models can also perform tasks like this, although there are many issues related to reliability and scalability that classic NLP systems suffer from less. Without going into much methodological detail, ultimately what happens is that Indra integrates across these sources in a way that we can take a specific mechanism, we also call it a statement, like RCHY1 ubiquitinates P73, and align all of the evidence supporting this statement from all of the different papers and databases that this was described in. Importantly, this gives you a sense of context, so it tells you where in literature, what kind of experimental context or disease context a given interaction was described in. And often for some of the most widely published statements, we have tens or even hundreds of thousands of pieces of evidence, something like P53 binds MDM2, hundreds of thousands of pieces of evidence from papers. Some of the applications of this, of Indra include these, so natural language-based model building, systematic explanation of experimental results from high-throughput data, human-machine dialogue systems that build on this knowledge base, as well as some community-specific data portals where we look at a subset of proteins of interest like kinases, the ubiquitinating enzymes, and create self-updating knowledge bases that you can go in and look at the latest results relevant to that given field, among other things. This is an example of what we call the rapid prototyping of mechanistic models, where you can describe a hypothesized pathway, hypothesized in the sense that you are encoding your assumptions about some mechanism in natural language, turn that automatically into a model using Indra, look at whether it reproduces what you expect to see qualitatively, and then if not, then you can modify your assumptions by rewriting some of the sentences, look at the implications of your modifications, and do this iteratively until you end up with a set of mechanisms that qualitatively reproduce what you are looking to see. Without this kind of automated approach, this would be a very slow process. A second type of application is it relies on more large-scale reading of literature and assembly. So in this example, we took about 500,000 publications and the content of 7 structured databases, built a network using Indra, a mechanistic network that has some resolution in terms of the types of mechanisms being mentioned, and used that to construct mechanistic explanations for data from the cancer gene dependency map. Interestingly, what we found was that the stronger gene dependencies in this data set are more likely to be explainable based on disassembled prior knowledge from Indra, indicating that certain things are well preserved across contexts and have been observed many times in literature. We scaled this up to all available literature and built a database out of it that you can query at db.indra.bio. You can browse the underlying evidence, submit curations if there's something incorrect. And also prototyped a way to turn this into an autonomous system deployed on the web, which, given a specific context, for instance, COVID-19, every day reads the literature relevant to that area, updates a model using this automated assembly technology based on new knowledge, can report on what it found, and then run relevant analysis like causal pathfinding in this model to try to explain experimental observations. Then it reports on new things that it found. So you can actually monitor publications in a model-driven or knowledge-driven way, where you look at not just what publications came out, but what the functional effect of the new statements in those publications is on a model's ability to explain observations. Going beyond causal mechanisms, we've extended INRA to a knowledge graph, which includes relationships that represent not just the basic processes that drive cellular behavior, but other types of ontological relationships, relationships representing data, things like clinical trials data, NIH funding data, drug side effect data, and things like that. And so this is available through an integrated knowledge graph through a web application, the endpoint of which is often analysis that you can run against data sets. So for instance, you can do causal overrepresentation analysis of upstream or downstream factors with respect to a set of differential proteins. And are able to browse the underlying evidence from literature that explain these interactions. And so last thing I will mention is that Olga Vitek's group, we are collaborating with them on integrating some of these explanation tools directly with data analysis software like MS Stats. So you can leverage some of these capabilities directly in the same place where you do your data preprocessing. So all this leads towards a vision that's kind of a cycle between experimentation and computational modeling and explanation. So you collect some data, you have AI-assisted annotation of that data to facilitate its deposition in these community data portals. Then there's a layer of AI-assisted data integration to pull out different data sets from others to leverage. AI-assisted extraction and assembly of knowledge and reasoning to ultimately generate hypotheses, which then help you design your next experiment. And all this is centered around a shared data fabric along the lines of what I described. And so with that, I want to thank members of my lab and the various funding sources that have supported this work. I'm happy to take questions.
Questions, and I believe there's a microphone in the back.
Hi, thank you. Really interesting presentation. So our group comes more from the environmental side. And so while we don't necessarily look at biological pathways, we look at things like environmental degradation products and different transformation products that form. So I'm really curious to know if this kind of framework and methodology could also be applied to environmental contaminants that, you know.
Yes, in many cases, conceptually, definitely. The practical side of it could require more work. I think the things I said about semantic technologies and integration, definitely applicable. It might require some amount of work to integrate sources that are not covered in the bioregistry, let's say, but are tailored to environmental studies, but conceptually the same can be applied. And in terms of the text mining and knowledge assembly side, I think one limitation is just that the knowledge extraction procedures and representation are sometimes tailored to mechanisms that are relevant for biology. However, a lot of it is actually at the level of generic causal relationships. So, you know, any kind of statement about something increasing another thing or decreasing it or association or correlation association will actually span across disciplines and not have to be necessarily adapted. So yeah, I think conceptually definitely applicable. Practically speaking, might require more work.
Okay, thank you.
Thanks. I'm very interested in how would you conceptually and also practically to use the AI to help facilitate the annotation. Currently in my organization, we feel like to extract the information, there are many ways, but if the information at the beginning is very noisy and inconsistent, annotated, it seems like increase a lot of like extra work down the road. So could you comment on that?
Yeah, so you're saying that you produce the data set and you want to annotate it in a way that it becomes reusable. Right, exactly. So I think the use of identifiers and ontologies and so on, that's something that helps with that, but it's very labor intensive to try to manually do that, and that's where various machine assistance approaches come in. So some of the tools that we've developed, you might be able to run them locally and find identifiers for all the drugs that you just have names for, or cell lines that you used, and so on. But that still requires some work. So where I see this going in the future is really these integrated AI-assisted workflows where you can talk to a machine assistant who in the background uses some of these structured tools to create that data annotation, but you're guiding it at a kind of higher level of interaction. The specific tools, if you go to my lab website, you can find links to a lot of the software, and we develop everything open source, so whatever is currently available you can find there. Otherwise, I'm happy to follow up and talk more.
Very exciting. Thank you.
Thanks, Ben. I think one part you mentioned in the knowledge assembly was imputing kind of missing information when it wasn't there. I was wondering what sort of missing information you normally see and how you go about actually imputing it.
Yes, very good question. So one example that I can say is, imagine, for instance, you have a kinase phosphorylating a substrate. Then there are at least 3 types of, or maybe 4 types of information that might be implied by that. One is that these 2 proteins bind to each other. 2 is that one phosphorylates the other. 3, that phosphorylation might be activating, right, or deactivating. So the overall effect of that, and potentially more. And very often what happens is that you only have a subset of these. So for instance, somebody might report that A phosphorylates B. Can you infer that they also bind to each other from that? That might not have been explicitly described. Or they say that A phosphorylates B and B on site X, and A activates B. You might infer that X is an activating site because of that. So that's the type of missing information that I'm talking about, things that are not explicitly described, so you don't pick them up, but you can infer from the things that you do get. But the interesting thing is, of course, doing this at scale, right? So for individual things, you can figure this out, but at scale, it would be more difficult to do. Yeah.
One last question.
Just out of curiosity, do you often run into like conflicting knowledge?
Yes.
Reports that are,
Very good. I get this question after every talk, so that's why I don't talk about it. Yes, absolutely. So in this database that I mentioned, it's full of conflicting information. It's not meant to be resolved at the level of the underlying database. Conflicting information can happen for multiple reasons. The most interesting one is when both the one direction of information and the other direction are true, and it depends on context which one is correct. And so we're interested in looking at analyzing the context, the biological context, the experimental context in which something was observed, to determine whether it's really this apparent contradiction can actually be explained by context or not. There are certainly genuine contradictions as well, so the same context but different observations. The point is that through a database like this, you can discover these and analyze them. And very often in our own end applications, you can actually leave both like two types of mutually conflicting information in what goes into your analysis workflow, because when you bring in data, which is always context specific, this learning process by which you adapt your model to data often naturally accounts for putting higher weights on things that are consistent with your data and lower weights on things that are inconsistent. And so often you can actually leave conflicting information in what goes into your data analysis because of that. So that's, yeah.
Well, let's thank Ben once again. Thank you, Ben, and I hope we will continue these discussions during the break. So our last speaker before the break is Wengong Jin, who is recently joined Barnett Institute as a faculty fellow and recently joined Khoury College of Computer Sciences as assistant professor. And Wengong will talk about accelerating drug discovery via geometric and generative AI.
Thank you so much. Thank you, Olga, for inviting me to speak here. Today I will talk about my work on accelerating drug discovery with deep learning models. So as you have seen, there has been a lot of excitement in an impact of AI to biology in different biological areas. And I am very interested in drug discovery and molecular and protein level biology, especially designing small molecules or proteins with therapeutic properties. And in this area, there has been a lot of excitement centered around protein design, AlphaFold, protein structure prediction. So these are actually very exciting era for computer scientists to contribute to the new biological discovery. So I'd like to kind of briefly motivate my work through this simple sort of narrative. Why do we need to accelerate drug discovery? It is because the wet lab experiments are generally expensive. As you can see from in the pharmaceutical industry, if you want to measure a property of a molecule, whether it binds to a protein or whether it is kills a cell, kills a cancer cell, you need to go through wet lab experiments. And in order to discover a drug, you need to screen through a large quantity of candidates. And of course, the theoretical, there's a lot of debate on what's the theoretical upper bound of the chemical space. They can be 10 to the 60th or beyond or lower, but it is a gigantic chemical space. So we usually could only test a very small subset of those. We would like to accelerate this process by not doing experiments for every possible candidate. We like to train AI models to do sort of virtual experiments. Now, instead of doing experiment for this molecule, we could train a deep neural network to predict its property as a proxy or surrogate of the wet lab experiment. And if we can do this well, then so in order to do this well, we need to train the model on a set of experimental data, but this doesn't have to be big. And we'll see from the later slides that we can actually succeed in doing this with just a small set of training data from the experiments. So basically, the general recipe is we train an AI model to learn this structure-activity relationship. The other way of looking at this problem is you could look at this as building a model that maps building a bridge between the two spaces. One is the molecular space, where all the possible candidates or molecules are, and we have the property space, and that characterizes where is the global optima of the molecule that you would like to use as the drug candidate. So I'd like to give three short vignettes centered around this idea of learning structure-property relationship and how to use that to accelerate drug discovery. So the first vignette I will talk about is virtual antibiotic screening. Here we discovered a new antibiotic for MRSA, a methicillin-resistant Ascaris. It's a very nasty bacteria where actually one of my friend's grandmother just passed away because of this. So it's very, very sad. But yeah, these antibacterial resistance is a really big problem. So the second vignette that I would like to talk about is generative antibiotic design, and where we actually, instead of just screening through these candidates, we like to apply rational design to generate candidate molecules, to design a molecule with desired property. And here we'll show preliminary results on a new antibody candidate for drug-resistant gonorrhea. The third vignette that we will switch from antibiotics to antivirals and anti-cancer drug combinations. And here we'd like to we'll show you how we can operate with very limited amount of training data, but utilize prior biological knowledge, similar to what Ben is working on to discover new drug combinations for COVID-19 and pancreatic cancer. So these are some of the summary of today's talk. So let's go through the first part on antibiotic screening. And here we, in collaboration with the Broad Institute, we collected phenotypic screening data around 40,000 drug candidates, 40,000 molecules with their growth inhibition against staph. Here we binarize the growth inhibition data into binary label, whether this molecule can inhibit the growth inhibition of staph or not. And so the basic idea of this project is we like to bootstrap a model from a small set of known drugs and extrapolate that to predict the antibacterial property of a large space of unknown molecules, and going from 10 to the 4th to 10 to the 9th. And the basic idea behind this is we train neural networks to predict antibacterial activity and toxicity. So at the same time, we collected training data that measures the cytotoxicity of this molecule in 3 different human cell lines. And we trained a graph neural network model to predict to train this classifier based on graph neural networks to predict what kind of molecules are antibacterial at the same time being non-toxic. So based on this, after this model is trained, then we can use this model to rank a large list of chemicals in the library, right? So and we can characterize 3 axes of each molecule. One is the predicted potency, whether it can kill the bacteria or not, and its cytotoxicity, and also its novelty, meaning the similarity to existing antibiotics. We definitely want to find candidates that are novel, that are not an improvement of the existing antibiotic classes. So here we discovered 2 candidates with the shared pharmacophore, which is highlighted in red here, with actually better potency than current first-line treatment for MRSA. And we can see that the discovered compound 1 and 2, we don't have a good name for those, but they show comparable, actually slightly better potency, and also good low toxicity. And we also looked at its resistance to resistance. Basically, if we treat the bacteria with this compound, we wanted to see whether bacteria could develop a resistance over a course of period of time. And this is a picture that we showed over. This is the change of MIC, the minimum inhibitory concentration or potency, of this compound after treating the bacteria over 30 days. And you can see that this blue dot, which represents our compound, does not show much loss of function, right? So you can see that over a course of 30 days, the bacteria cannot develop resistance against the compounds that we discovered. Whereas the standard antibiotic in clinic, you can see the bacteria can quickly develop resistance to avoid, to escape from the treatment. We also tested this compound against a wider range of bacterial species, against several resistant bacterial strains. And you can see that in general, of course, with different range of potency, you can see that our compounds are generally active against these different bacterial strains. So that's kind of one short vignette on how we can use simple neural network model to rank a list of candidates and use that to accelerate the antibiotic discovery. But of course, if you think about this, ranking is not necessarily the best idea. So here we'd like to illustrate the power of generative AI in antibiotic design. And here we're going to switch to a different bacteria called gonorrhea, which is a gram-negative bacteria that's a lot harder to treat than the gram-positive bacteria like staph. We similarly collected, through this phenotypic screening, we collected around 38,000 data points with molecules and their growth inhibition against the gonorrhea bacteria. So we utilized the virtual screening paradigm that we developed in the previous part of this talk, and we discovered a few candidates that are relatively okay, that has potency around 16 micromolar. And you can see that these are not at the best sort of therapeutic index that you would like to see, right? So the best compounds discovered from these virtual screening are usually suboptimal, because you are only exploring a subset of the gigantic chemical space. And in fact, it's only a very tiny chunk of the whole haystack that you are exploring. So in order to explore much bigger chemical space, you have to use more combinatorial optimization techniques to explore the chemical space that are not in the standard chemical libraries. So here we use generative AI to further improve the compounds that we discover through the virtual screening by proposing or generating analogs compared to from these two starting compounds. And here we developed a generative AI model, which will showcase in the next in a few slides later. How can we design different analogs? But here the workflow looks like this. So starting from the two compounds that we discovered from the virtual screening, these two initial leads, we applied generative model. We generated 20 million candidates of these analogs, and these are de novo, and we used the property predictor to again rank these two candidates and select the top candidates for experimental validation. And you can see that with this the help of generative AI models, you can design de novo compounds with much better activity compared to the initial leads. Here we can see that the potency to kill the wild-type gonorrhea improved by over 10-fold, and more importantly, this compound is also active against the resistant bacterial strains with low toxicity. So this is very encouraging because you can now apply these generative AI models to design de novo compounds, and you don't have to just stick with the chemical libraries constrained by your chemical vendors. And this is comparable to the first-line treatment in clinic, but with much better potency against the resistant bacterial strains. So we also tested the killing mechanism of this. We did some further study on the mechanism of action of this compound. We did some studies, and we discovered that this compound actually compromises the membrane integrity of this of gonorrhea, which is the outer membrane of NG. And we can also see that this compound is very specific to gonorrhea. So this is very encouraging because in general you can develop it's relatively easy to develop broad spectrum antibiotics because even soap can kill these bacteria. So there's nothing special if you can just come up with something like soap. But here we have a compound that's very specific to gonorrhea family, and we further studied its mechanism of action, and we found that this compound targets a very gonorrhea-specific gene called MAFA, which is which only exists in this Neisseria family, and it's particularly abundant in gonorrhea, and it's related pathogen called meningitis. And you can see that these two, the efficacy of this compound against these two species are similarly active. And so we are further studying the function of these genes, which is still ongoing. So I just want to do a very short sort of description on the machine learning part, because I'll be talking about a lot about the sort of results, just to showcase what the generative AI model looks like. This is a particular model that I developed called junction tree version autoencoder, which is a model that can generate de novo compounds. And here, what's interesting about this model is it tries to mimic how chemists design a molecule by reasoning about these chemical motifs. And here, basically, this model learns to stitch different chemical motifs together. In each step, it selects a fragment, a functional group, and learns to kind of iteratively expand the molecule until the model decides to stop, until the model thinks this molecule is effective against the bacteria. So this is sort of the rough idea behind how the model works, but I'm not going to talk too much on the mathematical details today. So without further ado, I want to move on to the third vignette that's a bit moving away from antibiotics, but reason about drug combinations. And of course, this can be applied to a variety of therapeutic areas, including antibacterials, antivirals, and cancer. Right? So just very quick introduction on what synergy is. We would like to find molecules that are synergistic to each other, that can boost each other's efficacy if we put them together as a formulation. And this has profound sort of success in the case of antivirals, especially HIVs. That nowadays the default treatment for HIV is all 2 or 3 drug combinations. So this work is developed during the days of COVID, when we were trying to think about how to develop drug combinations for COVID, motivated by the success of drug combinations for other viruses like HIV. So here we developed a model that tries to leverage biological knowledge by integrating multiple sources of data. And we argue that why do we have to do this? Because if you look at the 2 previous vignettes, we were operating on a decent amount of data with around tens of thousands, right? But here in the case of COVID, the drug combination data is rather few. We were collaborating with UNCATS, NIH, but they only had around 100 data points. And during the COVID time, it's really hard to operate in the lab and collect data. So with this in mind, we try to seek alternative approaches that integrates multiple sources of data with the biological knowledge. So we argue that in order to kill a pathogen, we need to find specific targets, and these targets are can be COVID specific or could be host targets, and we would like to reason about the efficacy of a drug or a synergy of drug combination by reasoning about how they interact with these targets. And there is a kind of general intuition that if 2 drugs target, complementary targets, they might be synergistic. Of course, this is not always true, but this gives us a kind of motivation on integrating these drug target knowledge, interaction knowledge, into machine learning models. So based on this idea, we developed this model called ComboNet, where it's a multitask training procedure motivated by this drug synergy formula called Bliss synergy, but also how the drugs interact with these targets. So the key idea is that we predict how the compounds interact with each target, and also based on that, we predict its single drug, the drug efficacy. And we also reason about how they interact with the target as a whole, as a combination, and from that predict its drug combination efficacy. And then we plug that into the synergy model, the Bliss synergy formula to calculate the drug synergy. So here we can integrate multiple training objectives using the drug synergy data to predict to learn about drug synergy. We can integrate more single drug efficacy data to supervise this single drug efficacy prediction module. And we can also reason about the drug target interaction at this latent space. So we, with collaboration with NIH, we developed this model. We evaluated on their internal data, and we found that our model achieved the highest accuracy despite using a very few amount of data. And using this ComboNet, we ranked over 10,000 drug combinations and tested the top 30 in a CPE assay. And we found that in vitro, at least, there are 2 drug combinations that are highly synergistic that were with combination with remdesivir. We further extended this idea to other diseases like pancreatic cancer. And here we tested this idea on a pancreatic cancer cell line, PANC1 cell line, again with collaboration with NIH. And we found that if we train the ComboNet model on pancreatic cancer data, then we can actually develop much better prediction models. And here, this on the right-hand side, you can see a top 30 true positive rate, which is the, so basically we selected top 30 drug combinations from the ranked list, and we found that 83% of them were highly synergistic in vitro, and this achieves much higher accuracy than NIH or NCAT's internal model called, that's only 53%. And we found one of the best drug combinations had a 10-fold improvement in IC50. So just to summarize today's talk, the key idea, if you just want to have this one takeaway message, is we can train AI models to do virtual experiments by bootstrapping from a small set of experimental data. And we showed its success in 3 different vignettes related to antibiotics and also antivirals. So in general, you can teach models to predict the property of a molecule and use this model to inverse design molecules of desired property. With that, I would like to thank a lot of my collaborators during my postdoc time and when I was a postdoc at the Broad Institute, where a lot of this work has happened, and would like to continue this further at Northeastern. So with that, I'd like to thank everyone, and we'll just pause here and see if you have any questions.
Great talk. I'm curious whether you looked to see if there was any evidence of a microbe making that anti-gonorrhea compound, like it's in some database somewhere, it just had never been screened for antimicrobial property?
So that's a great question. So the initial compound that we screened was in the standard chemical standard chemical library. That was never screened against any antimicrobial context. But the compound that we designed on top of that, which actually is substantially different, but share a small subset of the substructure, that is totally de novo. Yeah.
I guess a follow-up question. Could you imagine trying to sort of reconstruct something about the biochemical pathway involved in the synthesis of that molecule to see if there are organisms out there that might be able to create something similar?
Yeah, that's a great question. We haven't really done that, but I think the biological pathway that we were looking at is the MAFA, MAFB pathway, where it's a toxin secretion pathway. So gonorrhea is a very interesting bacteria that it tries to attack other bacteria by secreting these toxins. And this is the MAFA pathway is the toxin secretion pathway. But when it gets blocked, it seems like it actually gets killed. Maybe it's because the survival of these gonorrhea actually heavily relies on these competitions. You have to kill others in order for yourself to survive. But these are only hypotheses right now.
No, I mean, I guess it does seem like maybe evolution would have found something similar out there somewhere. We just haven't encountered it yet in our sampling. If it's
yeah, that's totally possible. Yeah.
One more question in the back. I have a question about how you test for, specifically for the drug combinations. How do you test? Or do you take into account how the drugs are metabolized, if they could actually be used in vivo safely? Like if they are metabolized with the same CYP enzyme, do you account for that? And also, I'm curious if you could elaborate more how you account for toxicity predictions in the first vignette. What do you look at when you look at the potential toxicity for the compounds you examine?
Great question. So I would like to say that a lot of these are sort of early discovery process. So for example, the metabolism question that you asked is something that we are trying to explore more, but we don't have a good predictor. We are developing that predictor now by collecting some metabolic data and also ADME basically. So we are actively doing on that, and once that model is ready, then we can integrate that into our workflow to further improve the safety of our compounds. Right now, the way that we measure toxicity is by looking at the viability of 3 different human cell lines. after we treat them with the compound. And we want to basically make sure that there's no cytotoxicity. But of course, toxicity can be much broader than cytotoxicity. So these are just a partial sort of view of what toxicity is right now.
I know that some different moieties on compounds are known to cause toxicity. So maybe like it's
yeah, that's already avoided because we excluded the structural alerts.
Okay,
and also these pain filters. These are already applied, but I think toxicity is much more complicated than we think. It's not just presence of one specific moiety. But
Thank you.
These are great questions.
Thanks for the great research. I'm interested in the third one, the drug combination. So when we proposing the combination, it seems like you rely on the drug target is already known by the single agent. Is that true assumption?
No, we actually do not assume that we know the target of the compound, but instead we utilize the drug target interaction data in Campbell to teach the model to predict the target of a drug while predicting its efficacy and its synergistic effects.
Okay. And then for the information, for example, not already available in the Campbell, for example, if there's some like omics type of the readout for the single agent, do you think is applicable to that type of the data set, like highly dimension for the each single agent, but we can train the model to apply to see some of the synergistic interaction between the two compounds, for example?
Yeah, that's certainly true.
Part of our program. And before we talk about the scientific part, I think we can really not talk about the Institute and about the medal without talking about Barry. And I would like just to spend a minute reminding everybody about the contributions that Barry had to our community and to the university. So Barry is the Director Emeritus of the Barnett Institute for Chemical and Biological Analysis, James Waters Emeritus Chair and Distinguished Professor Emeritus at Northeastern University. Dr. Karger had made major advances in understanding fundamentals of chromatography and capillary electrophoresis techniques and their applications, particularly in the fields of protein and DNA analysis. He received many, many awards, most recently the Lifetime Achievement in Chromatography Award in 2022. And so Barry established the Barnett Institute of Chemical and Biological Analysis in 1973, long time ago. It started as a collaboration with College of Criminal Justice, and then with the generous support of the Barnett family, the Institute really took off. And in 1985, Barry received the James Waters Endowed Chair Award. And so during the entire history of the Institute had been very flexible and agile in its focus. So it started as with some of the environmental sciences. Then in the 90s, it was supporting many of the activities of the Human Genome Project. And in the 2000s, when next generation sequencing came about, the Institute had a big focus on chromatography and bioanalysis. And so at all times, the focus of the Institute was not just on a specific technology, but on combination of technology and applications. And the key of all the work that the Institute did was forming interdisciplinary collaborations and collaborations between academia and industry. And despite all the contributions that Barry made, I would like to argue that the biggest contribution of Barry and the main legacy of Barry is the people and the community in the Institute. The Institute produced over 500 PhD students, postdoc and staff, many of whom have gone to distinguished careers in academia, in industry, and many came back last year when we celebrated the 50th anniversary of the Institute. So before I go any further, let's give Barry a round of applause. Thank you, Barry, for everything you've done. And now that I have been a director for slightly over a year, I have a completely new appreciation of the contributions that Barry had now that I have these really big shoes to fill. And so we continue the work that Barry started. We now are 14 faculty fellows from College of Science, Bioengineering, Bouvet College of Health Sciences, and Computer Science. Since my background is in statistics and computer science, the focus is kind of shifting somewhat to incorporate computational methods, as you have seen from some of the presentations that we have. And we also hope to continue the community efforts, and this includes collaborations and also training. In particular, there are 2 events that are coming up just in a few weeks. So April 28 to May 11, we have May Institute on Computation and Statistics for Mass Spectrometry and Proteomics, which we organize here on campus at Northeastern. And we have a variety of topics starting from, just understanding the data as they come from mass spectrometers, teaching basic or intermediate R, Python, etc. Ben is teaching a module on Indra and how to work with knowledge bases. And we also have what we call a future developers meeting dedicated more for computationally oriented people, where we have tutorials on some advanced topics and also research in some of the recent topics at the interface of mass spectrometry and computation. And the second event I would like to advertise happens on May 27 and 28, also here on campus. This is a single-cell proteomics conference organized by Nikolai, also in the spirit of bringing together the community and talking about cutting-edge research. And so among the events, a flagship event of the Barnett Institute is the Barry Karger Medal in Bioanalytical Chemistry. It started since 2004 and was officially named as such in 2013. And the goal is to recognize an individual for their outstanding achievements in the field of bioanalytical chemistry, and also bring on campus such an expert so that the entire community can benefit from their knowledge and expertise. And so I am delighted that this year Bernhard Küster is the awardee of the Karger Medal for Bioanalytical Chemistry. So let me now say a few words about Bernhard. So Bernhard is a renowned expert in proteomics and precision medicine. His work focuses on drug mechanisms, cancer biology, and personalized treatment strategies. Dr. Küster studied chemistry in Cologne and received a PhD from Oxford University. He had postdoctoral training at the European Molecular Biology Laboratory in Heidelberg and also in the University of Southern Denmark in Odense. After that, he joined the biotech Cellzome in Heidelberg, which is now part of GSK, and eventually became vice president of analytical sciences and informatics. In 2007, Dr. Küster was appointed full professor at Technical University Munich, and he served as department head for 13 years and Vice Dean of Information Management for 3 years. Dr. Küster is a member of the German Academy of Sciences. He is the advanced grantee of the European Research Council. He holds not 1, but 2 awards from World Human Proteome Organization, one in 2015 for the draft human proteome, and one in 2023 for the work on chemical proteomics. As you have seen from the earlier presentations, resources provided by Bernhard's lab are widely used by many, many labs and are of a tremendous value to us. Bernhard coordinates multiple research consortia. He leads Clinical Mass Spectrometry Center in Munich and also the proteome that feeds the world network of Bavaria. In addition to all the academic work, Dr. Küster co-founded 2 companies. And he also has a very substantial education effort. In particular, for many years, he was the co-organizer of the Brixton Summer School in Proteomics that I've been very fortunate to attend for many years, and I can tell you personally, this is a lot of fun. And so with that, it is my great pleasure to welcome Bernhard on campus and to celebrate your achievements and talk about your work. And I'm very grateful and glad that Barry is here in the audience also to hear about that. So now we have the medal that we can actually give to you. More photos with Barry as well.
You hold on for me for a moment. I'm on?
You can, I can hold on to it, or?
That would be nice. Oh, that would be very nice.
All right. Well, welcome to Northeastern.
Thank you very much. Olga, yeah, thanks very much for bringing me here. I must say this was quite a surprise, and I guess whoever was on the committee and put my name forward, they found good reasons for bringing me here instead of the many, many, many other people who would well deserve that kind of recognition. And but I would also like to say that even though Olga said that this is to recognize the achievements of an individual, I'm a firm believer in that no individual is anything without a strong team. So the thank you goes immediately also to my lab, of course, but also the previous position that I had at Celsum, without which none of what I'm going to show would have happened in my career. And the great proteomics community as a whole, with whom we can fight at times, but also laugh at times, and hopefully move science along in the right direction together. And so this team effort that has been very important to me in any leading position that I've had is still important to me, and my lab doesn't hire anyone without the thumbs up from everyone in the lab. I will not run a project that people think is a stupid idea just because I like it so much. So thanks to a lot of people that helped me kind of go places, and it's in the growing family, both the current lab and the many, many grad students and postdocs who've been traveling along that trajectory with me. And of course, money is a necessity, but Germany isn't particularly known for big science funding, but sometimes by thinking about something more carefully, and if you really want to do it, you can also run bigger things sometimes with a small budget. If you have people who really set themselves to cracking a nut, it can often be done. So brain power is important, so thanks to this team. And to what the dean said earlier this morning, we need to all learn to say things in simple terms. I'm not sure that I will accomplish this fully, but I think this slide here is an important one that I would like everyone kind of to take in terms of why we're doing what we're doing. And we apply proteomics to cancer research and drug research. And why do we do both at the same time? Because history teaches us if we know what is causing a disease, we may find a medicine. And on the proteomic level, if you like, this could be either a biomarker or a target themselves, but it is nothing without finding that piece of chemistry that would go along with it so that the two together can enable a diagnosis and a treatment. And what we've been trying to do in the past several years is trying to broaden kind of the perspective on both of these angles in a way that says, can we use proteomics to learn more about the drug mechanism of action? What do they actually do? And can we also use the same technological approach to look at cancer patients and particularly trying to find what is driving, potentially what is driving those cancers? And can we bring the two together to have highly active signaling pathways in a cancer patient, and by knowing what drugs can do, and we're particularly interested in the repurposing idea of drugs, is can we bring the two together in order to kind of provide potential treatment options for people who are out of other options. Okay, so, and the justification why we think proteins are a good place to be when it comes to study them, this is a map that a group in Arizona nicely puts together every year. 3 years ago, it would have been full of vaccines of the mRNA type 3 years down the road. They've all gone, we're back into the small molecule and antibody world as therapeutics. But I think the takeaway from this slide is all drugs act on proteins, are proteins, make proteins, degrade proteins. And that is even true for mRNA vaccines. Okay, so proteins is the molecule to study when you want to look at drugs. And one has to realize that drugs typically have more than one target. Even though the bottle that you use in the laboratory says, I'm a potent and selective CDK9 inhibitor, I totally buy the first part of the sentence. I totally don't buy the second part of the sentence. So many, many drugs can do more than one thing, and that can be a good thing. It isn't only just about tox. Okay, so what we try to learn more about is, can we systematically understand what they do and then repurpose them for useful other things. And so this is why the lab is structured in the way it is. On the left-hand side, there's a team of people who looks into these cancer proteomes. There's a team that tries to understand how drugs work, and that's kind of the focus of today. But at the same time, we always need better tools, and a good piece of the team is also developing both laboratory and informatics tools to do the other two things better every year. Okay, and you're going to see this slide a couple of times, because I'm trying to convey a very simple concept that is not even very original. It certainly isn't ours, but it's a very good guiding principle. All drugs act in a time and in a dose-dependent fashion. And the dose dependency part is obvious to many of you, but it's often overlooked. And this is going to be the recurring theme here. Everything you're going to see from here on is going to be based on dose-dependent measurements. And that is an important part, because if you think about such a prototypical kind of potential oncogenic signaling pathway where you have, we can't point, can we, with this? It's not a pointer, I guess. Anyway, so you have receptor tyrosine kinase, for example, EGFR, at the top of the signaling cascade, and there's a small molecule drug that would, in a dose-dependent fashion, inhibit the activity of that enzyme. And on the left-hand side, you can see a dose-response curve for EGFR binding, and finding out which targets a small molecule has is important. But this binding of the target may not do anything unless the adapter proteins below this and the enzymatic, often the kinase signaling cascade, gets engaged and therefore also in turn inhibited. And then the cell at some point is going to recognize that what it is driven by, the signal that it is driven by, is cut away, and it may decide after some time to react to this, either die or compensate by some mechanism. And all of those mechanisms that follow from the application of a drug to a biological system, they all should be dose-dependent. And this is really important. And I put this slide in here because modern LC MS technologies are, in a way, the way to measure all this. Because it's highly quantitative. Yeah, so the signal that comes off from an LC-MS experiment is very, very quantitative. And we can measure lots of proteins, of course, at the same time. And that little formula that you see here is nothing but just a regression that describes the dose dependency of drug treatments of any kind, and where on the y-axis you have the response of the system, and at the y-axis you have the concentration of the compound applied, and then the dot in the middle is the effective concentration at which 50% of the effect has been achieved. So you get essentially two pieces of information: how much happens at what concentration. Okay, that's the basis in a way for all pharmacology. It's been neglected in proteomics for the most part, and I'm just hammering that into your brain. Never do a drug proteomics experiment again in your life without a dose response. Okay? All right. Because of the title of the award, also I said, let's put some analytical chemistry in here. And again, something very old and simple, but in the realm of proteomics, in my opinion, still very relevant, is we've all grown up with nano LC separations and mass spectrometry as a readout for the simple reason that it is very, very sensitive. But actually, we shouldn't forget that chromatography works along various flow scales, and you can actually make proteomics work at any of those flow scales. So what is shown here on the picture is, okay, an HPLC system, but 4 different column dimensions. One of them at the bottom is the typical 75 microns that everyone is using, and the one at the top is a whopping 1 millimeter column inner diameter that nobody in their right minds would use when doing proteomics, unless you realize some simple things that you can do nice chromatography at higher flow rates. Okay, and this is what we've actually looked at systematically with the idea is which proteomic application would go well together with what kind of LCMS because of the quantitative performance, the throughput performance, the robustness of it, and so on and so forth. So in what is here systematically described or compared is just different flow regimens on different sized columns using exactly the same sample and exactly the same injection to injection time. And I'm not expecting you to take in all of the data in here, but I think suffices to say, depending on how much protein you have available to your analysis, you don't always have to use nano. And often a good compromise is actually good old capillary flow, LC separations, because they often come with better chromatographic characteristics than nano separations. So here, when we first look on the right-hand side, what is plotted is the chromatographic peak width as a function of these different flow rates, and we can see that at higher flows, the median half-width chromatographic peaks are just much narrower than on the nanosystems, and that has the effect that you can compensate a lot of the loss in sensitivity that you go at higher flow rate by having sharper peaks, getting higher concentrations, getting more signal. And yes, the numbers on the left-hand side just compare the sensitivity difference to 50 microliters a minute, and you could see that, well, sometimes the increase in sensitivity is ginormous with 2,000% when you go to nano. That's not new news. But if you take a spot of blood from someone, and just from a prick in your finger, you don't have to worry about 100 nanogram of protein, okay? You have more protein in that one prick test that you'd ever want to analyze. So why use a finicky nano LC system if you can run down your sample with 50 microliters a minute? And we've done more than 100,000 proteomes that way. So I get asked a lot the question, do you really use this or you just publish on this? And I said, well, after 100,000 proteomes, I think we can say we really use this. And quickly, just here, when you look at the right-hand side, these higher flow rates come with one particular benefit. It makes the whole system extremely robust when you want to run large campaigns of samples. And the quantitative precision here is expressed of coefficient of variation is just better than any of the other flow rates. And none of this is new. None of this is new. Chromatography people, of course, have known this before I was born. But actually, it's not really used much in proteomics. So what I'm trying to say by this also in an audience that cares about analytical chemistry and measurements, pick the right system for what you want to analyze, and don't look at every problem as if it is a kind of nail because the only tool that you think you have is a hammer. Okay, I think that's kind of the bottom line. And we could say the same thing about statistics. Which brings me back to this slide, where we do a lot of dose response measurements, and I asked my PhD student, there's got to be a way of treating this statistically so that when we are going to generate millions of dose response curves, we can't look at them, all of them. I mean, human eyes are very good at looking at dose response curves and figuring out whether there's something there or not. And, but if you have to do this tens of millions of times, not really working. So what he has figured out is the maths that is beyond me, but I'll walk you through the Bernhard simple thinking type of style. What is wrong with that experiment? So here we have a cell culture system. We treat with an EGFR inhibitor of 10 micromolar. Do this in a couple of replicates. You send it down your favorite mass spectrometer, be it TOF-based or be it trap-based, doesn't matter. And then you do your t-test, and people are happy. So what's wrong with that experiment? I could ask. Nothing per se, you might say, but all the white space here I blanked out simply because it kind of focuses your visual attention on what we think is going to go up and what is going to go down. And who's to say that cutting the PL value axis where we cut it or the fold change axis where we cut it makes any sense? It's very practical, but you don't know absolutely anything about at which concentration the drug actually did something. Okay, so you might say, then let's do that experiment. And it's a real experiment, and my PhD student did all the experiment I told him in the beginning until he saw the effects. Let's do everything in triplicate and compare afatinib at nothing dose to nothing dose to 30 picomolar, 300 picomolar, 1 nanomolar. And you can see the volcanoes here. None of this is happening, nothing is happening anywhere. And now where the cellular activity of afatinib is at about 30 nanomolar. And all of a sudden your volcano plots start to show blue and red things. And at some point we end up with 10,000 nanomolar, 10 micromolar, and then we have hundreds of things. Same problem. How do we know that any of this bears any significance other than the t-test? So the better experiment to do is actually to do this in a dose-dependent fashion, because then your volcano plot, when you drop the p-value from the y-axis and plot the negative log of the EC50, which is the inflection point of that dose response, the data now becomes a lot clearer. So you have a big red blob of cases. All of these are phosphopeptides, by the way, I should have said this, at around 10 nanomolar, which actually happens to be the interaction constant of afatinib with EGFR in a cell, which then kicks down to all of the phosphorylation sites that follow exactly the same dose-response characteristic. And everything that is down at the bottom of this 10 micromolar or 1 micromolar, even 100 nanomolar, is all completely irrelevant for the mechanism of action of this drug. It's just all kind of bystander side effects, secondary things that have nothing to do with what the drug is actually doing. Okay, so, and from that, FlowBio built a little tool that is called Curve Curator, which tries to find the best kind of fit through the dose response data and then apply some F statistics. And I think to just cut this short a little, with the intention to be able to distinguish kind of the 4 cases on the right-hand side. Dasatinib and CSK, it's actually a target of that kinase inhibitor. And when you look at this, the Curve Curator output basically tries to figure out whether a dose response is significant statistically, and it also gives you the size of the effect. So on the top curve it's obvious, yeah, so the red line follows the dots, the measurements perfectly. So no wonder that this is a clear case of not only you get an EC50 value, but it also comes with high statistical power. But then the second case is actually one of the brutal killers for us when you make millions of these curves. If you do a normal regression, you always get a fit, and the R squared of this is of course perfect because the data is good. But it's a completely insignificant regulation because there's no effect. Okay, so this is why the p-value here on the second case is actually poor, because there's actually almost no measurable effect. And then the third case is, well, there's one outlier data point. For whatever reason, that data point is not good, and that gets penalized by the significance test simply because of that outlier behavior, even though the total effect size appears to be big, but we can't be certain that this is something there. And the fourth case here is now both things happen. The data is just all over the shop, and you really never want to spend any time looking at this. Okay, so these detours were purely meant to say that good measurement and a reasonably simple type of data analysis are going to help us a great deal when now going through a lot of proteomic data. And the next slides are going to be perhaps too laden with data, but if I want you to remember just one thing from this, doses, responses rock, and they do help you in any and all circumstances. Okay, because it also has biological consequences. So here, when the output, or when you follow the Curve Curator idea, you can also get volcano plots like here on the left-hand side, where now the y-axis is not a t-test p-value, but it's actually what we call a curve significance. Is something happening? And when you plot it again in the middle with the PEC50 value, you very clearly can distinguish, and I said that before, those cases where the drug treatment actually inhibited the target and engaged the downstream pathway. And these are just the molecules at the bottom in the legend that are known factors in EGFR receptor signaling. And everything, and the pink line in the middle is where cells die at that dose of the drug applied. So, and then the orange dot in the cloud further down is P38. It's P38 MAP kinase phosphorylation. It has nothing to do with that pathway. So we can relegate all this away and focus our attention on those things that actually matter. Good. So that was on the technology, and I probably have used much more time than I wanted for this, but I felt it was important to say a few things about these, because what we try to do is, when we do these experiments, we approach them in a relatively simple way. Conceptually, we're trying to do good measurements and then use simple analysis ideas to try and derive some information from it. And in the chemical proteomics world, for me, it started with the guys on the left-hand side, Markus Banchev, who's just moved to Roche, and Gerard Reves from Cellsome, where we started to think about chemical proteomics in 2002, and the rest of the faces here are all the PhD students and postdocs who have worked on topics related to that since 2007. So I'm super grateful to all of these people for spending years of their life trying to pursue an idea that says, can we use proteomics to learn something about drugs? Okay, so let's walk through this. Target deconvolution. First step often that we want to know in a bioactive compound, what is the target that it hits? And for kinases and HDAC inhibitors, we've gone down a very simple kind of experimental paradigm where we do affinity purifications using derivatives of, in this case, kinase inhibitors. They are very, very broad spectrum, so they can bind a whole target class. And we can put them on beads, and we can use those immobilized kinase inhibitors to pull down kinases from any biological system that we want to, cell lines, organoids, even tissues of model systems. And then the idea is, and that is essential, and this is why that paper ended up in, the original paper ended up in Nature Biotechnology in 2007, is you introduce the idea of a competition binding element. So you throw your compound of interest into the soup of proteins, let it equilibrate out, find its target at whatever concentration it does so. You then throw in the beads to mop up all the molecules that haven't been bound by that free compound in solution. And then you're going to send all of that down a mass spectrometer and just measure the relative signal difference between competitor and no competitor. And if you do this in a dose-dependent fashion, you get those dose-response curves that you see here at the bottom. And one can actually show mathematically that the inflection point multiplied by a constant, what we call a depletion factor, is actually equivalent of a KD. And you get that not only for one protein, but for all proteins that can bind to that bead system. And the heat map on the right-hand side is now structured in the way that we've done this for 200-something compounds, all clinical kinase inhibitors at the time, and then ask the question which protein kinases get hit. And the color here in this heat map is now not an up or down regulation, it's actually the negative log of the EC50 value of that dose response curve. Okay, so there's 500,000 dose responses in that experiment, and the colors code here just shows you what does the compound hit, which protein does it hit, and at what concentration do they hit. And it makes a huge difference whether you hit something at 10 micromolar or 10 nanomolar. 10 micromolar you may never achieve in a person, or not for more than 2 minutes in that person, because the liver is taking care of metabolizing the molecule, et cetera, et cetera. So the interpretation of what this means is then important. And what it allowed us to do is to answer a question about drug selectivity. And most kinase inhibitors, and that was kind of in my opening remarks, are not very selective. However, I would argue that knowing what the selectivity is actually important, because it allowed us at the time to repurpose a drug called cabozantinib, which was actually made against VEGF receptor and MET at the time, and it's an approved drug, that we found completely unexpectedly that it is also a very potent FLT3 inhibitor that is driving some AMLs. And the mouse experiment here is just to show that when you inject these AML cells into mice, they very quickly grow in that mouse, but you can treat these mice with cabozantinib and effectively keep that tumor growth in the mouse down. So with this data package, you have enough information to go straight back into a phase 2 clinical trial now by switching the indication using exactly the same drug. All the in-human tox is all known already, and it actually turns out it is a better AML drug than it is a thyroid carcinoma drug. Okay, and then one thing that is why this is also so much fun to do is we always run into a lot of surprises. And here's another surprise. All lipoic acid. or lipoic acid. People have been wondering what it does, and people have, because of its chemical structure, thought about the reason why it is in a food supplement, and it could be good for treating kind of reactive oxygen species, is because of these two sulfhydryl groups. But actually what turns out when we looked at this molecule using beads that were carrying HDAC inhibitor molecules, it actually turns out that it is an HDAC inhibitor, but only the R form is an HDAC inhibitor. The S form doesn't touch the HDACs at all. So after years and years of kind of promoting lipoic acid as a food supplement justification for ROS, keeping ROS at bay, this is not at all what it does. It does reduce the stress granules in cells by way of inhibiting HDAC 1, 2, 3. Yeah, so it's a completely different mechanism of action than people originally thought. Now, these degrader compounds, PROTACs, are a big thing these days, and it turns out that proteomics is actually also a perfect way of looking at these molecules. And what you see here is one of those PROTAC molecules where you have a kinase inhibitor called dasatinib as a warhead, and then some linker molecule here, and then thalidomide as an E3 ligase recruiter. So it's a classic design of one of these PROTAC molecules. And what you can do by proteomic measurements is you can actually measure what this molecule does to your protein in terms of whether or not it degrades it. And the simple twist here is you can grow cell cultures under whatever normal conditions, but then you do what we call a pulsed Silac experiment. So you pulse them with stable isotopes, so then the stable isotopes gets incorporated into the newly synthesized proteins at the rate of the protein synthesis. And to really kind of prove that your degrader molecule is actually degrading the protein of interest is that you have to show that it removes the mature protein, but also the nascent protein. Because if it only removes the mature protein that was already there before you applied the PROTAC, it could also be transcriptional downregulation. And this is what happens a lot, that the cell decides to no longer make the protein, and then because of natural turnover, the protein quantity just goes away. So on the left-hand side, where you see that on the protein kinase called GAC, you can see a dose-dependent decrease in the amount of GAC, both the mature, the old protein, as well as the nascent protein, and the different colors of those lines just indicate the different time points at which these experiments was done. And on the right-hand side, and that's the surprises that come out of these experiments, ILK is also a target of dasatinib, but you can see that neither the mature protein nor the nascent protein actually get degraded. So the specificity of these PROTAC molecules are driven by more than one thing. It's not just the warhead, it's also the part the E3 ligase recruiter, and in fact even the nature of the linker. So, and without mass spectrometry, this is very hard to measure, and a very exciting, in my opinion, application areas for dose-dependent measurements. Cysteine covalent drugs are also a thing. Same thing applies. You can actually, and Steve Gigi has done a lot of that work here on campus, where you can fish out cysteine-containing peptides from protein mixtures, and you can do this by treating cells in a dose-dependent fashion with this molecule that you see here, which is a covalent modifier of KRAS G12C. Yeah, so it's a KRAS mutant, and that is driving quite a lot of cancers. And actually, when you look at this, the bottom left-hand corner, out of 12,000 or even 20,000 cysteine-containing peptides that one can measure this way, there's only KRAS G12C that is being inhibited by that very molecule. So it's actually, when you make these covalent drugs, a super high content assay to ask the question, what other cysteines chemically react? If they do so, at what concentration do they react? And if you find that one of these cysteines get modified at 10 nanomolar and the other only at 10 micromolar, you know that you have a 1,000-fold therapeutic window over hitting that target versus the other. So returning to this one, and now looking at the next part, which is the engagement of the signaling pathways down the road, you would expect that if EGF receptor signaling powers, let's say, the proliferation of a cancer cell, you would not only have high activity of the receptor, but you will also have then high phosphorylation of the transmitting signaling pathways. And we can measure this in response to drugs again. And we've done this now at the level of the post-translational modifications, because kinases phosphorylate other proteins. Conceptually, you could do the same for ubiquitinylation or acetylation when you want to look at HDAC inhibitors or CAT inhibitors. And if you were to take your phones out and wanted to look at a few million dose responses, here's the QR code that takes you there. And the application, one of the applications that is quite interesting is you can generate kind of signatures for your compounds of interest. Here we have 10 kinase inhibitors on the left-hand side, and the distribution plots just simply say, or the numbers say how many phosphorylation sites go down in a dose-dependent fashion, and the x-axis is the negative log of the EC50, so the potency with which that happens. And you can then, the mother of all kinase inhibitors is staurosporine, so it inhibits many over a very broad range. But you can see that already some of these molecules are more potent, and some of them are more hitting more different phosphorylation sites. It's all in the same cellular background. But perhaps what is more useful to look at then is the right-hand side heat map, where every blue bar is again a dose-response curve of a particular phosphorylation site in response to, in this case, either AZD, AD55, or any of the other drugs. And you can see from a mile away that if we only focus on doing this for 10 drugs, most of that matrix is rather sparse. Thank God. There is some specificity in the targets that these molecules inhibit. But you can see there's partial overlap and partial non-overlap. And for example, the first 3 molecules, they are all known to hit mTOR, and then you can, to the extent possible, annotate these phosphorylation sites using phosphosite plus. So if there are any phosphosites that are annotated to be mTOR targets in there or substrates in there. But you can also see that some only partially overlap in the MAP kinase world. And I'm pointing out 2 protein phosphorylation sites in particular. One is BRCA1 and this ZX. Because the second, so the use case of these kind of barcodes, if you like, is you could look at them as a pharmacodynamic biomarker. What does the drug do to my biological system? And it's not one thing you measure, you measure a whole barcode of things. And if you imagine if you had this now 1,000 times, you could work out what the selectivity and specificity of these drugs are. But what is very exciting about this too, in my opinion, is you can start to give function to individual phosphorylation sites that people didn't know what they were doing. And that is the example of BRCA1 and ZX. Of course, the BRCA1 phosphorylation site is well known. It responds to a drug called dactolizib because it's down the road from ATR. And then ZX, phosphorylation site 956, nobody knows anything about, but it also has this SQ motif that is kind of a hallmark of ATM, ATR, and PRK, and DNA-PK kinases. And by the fact that they behave very similarly between different drugs, you can make an association of this phosphorylation site with this particular kinase motif to the upstream kinase that likely phosphorylated it is. That's inference, no proof, but it helps to put at least some label on all of these phosphorylation sites that PSB doesn't know about and we don't know about. And that situation is kind of reminiscent of DNA sequencing, where the DNA sequencing people have no problem recording all these mutations, but nobody knows what most mutations that are detected in humans mean. Is it disease? Is it no disease? Is it just natural variation? We don't know. Same goes for phosphorylation. There's almost nothing we know about protein phosphorylation so far. And you could start putting some functional annotation into this by way of the drug response that they show. And to the point of synergy, here's a PhD student, Steffi Höfer, that has just come out. We asked the question, can you actually combine 2 different drugs to get synergy? And here she did this on 13 cell lines and 150 mostly kinase inhibitors. And actually, interestingly, if you just look in the heat map on the far left-hand side, there was one drug that seemed to show synergy in these 13 pancreatic cancer cell lines in combination with gemcitabine. So I'm very grateful to the talk on the AI part earlier today, where what you don't see on such a slide is what we have in pictures when Steffi was at the EMBL screening facility, and the towers and towers of plastic that she was standing in by just screening 13 cell lines against 150 drugs in a dose-dependent fashion and a couple of replicates. Yeah, so these are fun pictures to look at. And you boil it down to this, so if AI can help us to do a fewer experiments, we'd love that. There's too much data on this slide, but I'm jumping over this. What comes out if you do the dose-dependent measurements of what the phosphoproteome, how the phosphoproteome reacts to an insult by gemcitabine, which generates DNA damage, and the cell responds by upregulating its DNA damage repair machinery, which is governed by a kinase called ATR. And if you then kick in a drug that inhibits ATR activity, you get drug synergy on a phenotypic level. And we can actually, from the molecular data, pinpoint that it is all just about ATR. This is a screenshot from PTM Navigator, so a tool another PhD student in the lab generated, is where all the regulated phosphorylation sites, you can see they are more on the left-hand side around ATR as a kinase, and nothing is on the right-hand side on ATM kinase, which repairs a different type of DNA damage. And yeah, molecular systems biology was nice enough to choose our suggestion from an AI-generated picture of gemcitabine and elimusatib breaking into the fortress of pancreatic cancer and giving that cancer a hard time. Okay, I need to look at my chairwoman to make sure I don't go completely crazy. And also in these experiments, more often than not, you run into surprises. A couple of years ago, I visited Genentech and I asked Vishwa Dixit, do you actually know how rituximab works? So rituximab is the first ever approved antibody to treat B-cell malignancies. And it's still successfully used today for depleting B cells, essentially, in either a cancer environment, but also for multiple sclerosis. And the answer 5 years ago is, well, we know it works, but on a molecular level, we really don't know so well how it works. So we thought, okay, let's throw rituximab on some CD20 positive B-cell lymphoma model. And the idea is that rituximab does bind CD20, and it does so, and then the cells die. And the question is, how do they die? And there was no good answer from that, from Genentech. Well, the immune system probably comes along, so there's this classic case. So we looked at this, and what we actually saw, and these are the dose responses that you see here, is we saw phosphorylation of SYK and BLINK and MAPK14 and MAPK1 shooting up in a dose-dependent fashion in response to rituximab. So and on the right top corner is a protein called raftlin-1. It's the major protein constituent of lipid rafts that are floating on the surface of cells. And their phosphorylation went up massively, but also this is in different time points. So at short time points, it shoots up, but after some hours, it also gets kind of toned down again. And this is now 8 weeks of frantic working with half the lab when we threw rituximab plus and minus different kinase inhibitors onto that system to work out which of those 5 major axes of B-cell receptor signaling are affected by rituximab. And the reason why I show this complicated slide is because it is complicated. B cells, some of you probably know this, are constantly in need to decide, am I going to proliferate and make antibody? Or should I die and get selected against so that my immune system is kept in check? And those signaling pathways are the ones that keep these B cells in balance. And what seems to happen is that the binding of rituximab to CD20 activates the B-cell receptor signaling pathway, and it does so by hammering down the MAP kinase pathway. So you get disbalance between those signaling pathways. On the NF-kappa B side, mTOR side, nothing happens, but on the P38 MAP kinase, classic RAS MAP kinase, and calcineurin, calmodulin pathway, phosphorylation all goes up strongly, which basically makes the whole system out of balance, and then they slip into apoptosis. And we've repeated these experiments now in other B-cell systems, and we see the same thing. So the surprise now, after 25 years of clinical use of rituximab, is there's now a much better understanding on how that actually happens. There's no change in clinical practice, of course, but it's good to know. All right, and then the last step in this is taking it down to the last part. Let's say when the cell senses that perturbation, it generally reacts, to a perturbation, if that is life-threatening at least, and then it has to kind of decide how it may change its cellular program, expression program, to adopt a new phenotype. Or if that can't happen, then to die. And there's an exceedingly simple experiment you can do. It sounds complicated, or looks complicated here, but I haven't really shown any workflow pictures. But this is how it looks like in the lab. So you can just throw compounds onto cells, onto live cells, and let them incubate for a day or so. And then you can just ask the question, does the cell change their protein expression profile in a dose-dependent fashion? Yeah, and not just yes or no. And so two students, Stefan Eckert and Nikolai Bernhard, have done that experiment, and we published a pilot last year, and they've now done this for 2,700 drug molecules, which two students and one SMA Bravo could actually handle by just incubating, in this case, Jurkat cells, in a dose-dependent fashion, wait for a day or so, and then you do automatic SP3 digest, and then we send it all down a single mass spectrometer, and to record then the expression changes in response to these drugs. And that's another 17 million dose responses. So, and what is interesting when you look at this from a distance, here the main figure here is just plotting how many proteins go up and down and sorted by drug. And on the top right-hand side, the inset is that the same plot after 2,700 drugs. So you see a wide range of things happening, either going up or going down. And that was unexpected for some areas, and some of them are completely expected. Let's say on the left-hand side, you have things like proteasome inhibitors. So if you inhibit the proteasome, you screw up the proteostasis of a cell big time. So everything accumulates, lots and lots and lots of things get not upregulated, but actually stabilized because the proteasome can't get rid of them anymore. Or an HSP90 inhibitor that needs to fold just about every other protein in a cell. If that can't function anymore, it's a major insult to the proteostasis of that cell. But then there are other compounds that do a lot less and sometimes upregulate things only or downregulate nothing or the other way around. But the nice thing about this is behind those histograms are now dose responses, so we can get an information about how much and at what dose do these things happen. And for a selected number of compounds, and I'm not a big fan of transcriptomics, but for this experiment we did some transcriptomics just to ask the question, is it transcriptional downregulation or what is it? Is it stabilization? Is it degradation? What is it? And here's just 5 proteins, or 6, I'm sorry, where you can see for HDAC1 expression in response to vorinostat, neither RNA nor protein level changed, even though the enzyme gets inhibited. It doesn't necessitate, there's no necessary causal relationship between inhibiting something and then making it change its expression. Palbociclib, different story, both go down, which means the cell downregulates on the transcriptional level, has nothing to do with protein level regulation. The cell just runs dry after a day of not making any more of these RM2 proteins. Carfilzomib, proteasome inhibitor, the mRNA shoots up, but the protein doesn't really. So the cell somehow senses it can't degrade proteins anymore, so I need to make more of that machine, but it's a futile exercise because that machine is being inhibited by a small molecule. And the other way around, you get, I'm jumping to the last slide because it's a classic molecular glue, pomalidomide, where the mRNA levels really don't change much, but the protein disappears. So there's active protein degradation happening by that drug. So all of these possibilities exist, but the initial experiment to do is quite simple to do, and to get kind of an idea of where you should be looking. And when you put these all these data together, again you get a very sparse matrix because most of these molecules don't do anything to most of the proteins, and then because we selected drugs from all corners of pharmacology, we didn't necessarily expect that they would form strong functional clusters, but some clusters you can see, and the cases that are kind of boxed in with cluster 1 and cluster 2, and they kind of look like mirrors. One is going up and the other is going down. And essentially what this is, you can see in panel B here, all of those drugs, in fact, arrest cells in the cell cycle, but they do that at different places. So the G1 checkpoint is where cells stop in response to ulixertinib or nintedanib or some others, and the G2M checkpoint is where cells stop in the other case. So you could actually deconvolute by in this case, these are not expression heat maps. They are now actually Go enrichments of those proteins that show a dose-dependent change in their expression. And that is how we can bring them together on the biological processes and try to see if they affect similar or not so similar biological systems. And the usefulness of that is beyond being able to show that you can do this is, for example, pharma has phenotypic screening assays going where you don't learn much other than a similar phenotypic response. But if you've done a screening campaign of hundreds of thousands of compounds, you can then select individual molecules from these different groups and then do a simple proteomics experiment to get an idea of which biological processes may be affected. Yeah, that only works, of course, if your phenotypic assay isn't just kill the cell. That is an obvious readout. Now, more surprises, and we found in these experiments, or this basically Stefan and Nikolai came across the fact that HDAC inhibitors completely abrogate T-cell receptor expression, both in cell lines and in ex vivo, when we basically isolated T-cells from healthy individuals. I didn't want to know who that was, but I was told it's been done by the book. And completely unexpected. Why should an HDAC inhibitor remove the whole T-cell receptor machinery from the surface? And then what actually functionally happens is you can no longer activate those T-cells. And the reason why this is interesting, and perhaps important even, there's an ongoing clinical trial where the trial tests the combination of immune checkpoint inhibition with HDAC inhibitors. And according to our data, this is a really dumb idea, because you're trying to release the brakes from the immune system, and then you castrate the T cells by removing the T cell receptor. So we strongly anticipate that this trial is going to be completely and entirely negative, because these two drugs actively work against each other. So I have no idea what the original rationale was, but we don't think it's going to work. Okay, I probably have 5 more minutes or so. It's been said, thanks Nikolai for bringing up the point that other people are using our data. At some point, some years ago, we realized there's absolutely no way that we can work up all of the things that are potentially in this data. So we thought there's no risk in sharing. When I had been a much younger PI, we would have had a different idea about this. But what we do realize is no one of us, proteomics people are so data rich, it's beyond belief. So we can share. Should not ever be a problem to share, because, and tomorrow there's more. And we're trying to, at least the studies that are on a bigger scale, or where we think we're adding a new kind of concept to the proteomics type of measurements, we try to put them into Proteomics DB, and Matthias Wilhelm has kind of been the person who has led this effort from day one, and has got his own professorship now, so it's no longer in my team. But we still do put a lot of data into this system, because many years ago, already in biotech, I basically said we shouldn't approach every new experiment as if we had never done one before. So can we please learn more from the data that we already have? And database systems like that are one way of doing this. So if the semantics can be worked out, then this is good. By the way, you can pump this by APIs to your heart's content. Okay, so in the last 3 minutes that I have, and I'm going to be very quickly, I've only shown really the drug side of things, and I chose to do this because I thought it was a good fit to the Barnett Institute and what people do here and the whole mission, but I don't want to kind of leave without circling back for why do we even do this, and the why is also on the left-hand side. So we want to learn more about how we could use those drugs to treat patients on the basis of knowing much better what the individual cancer might be driven by, and that is what we call the TOPAZ idea, the tumor proteome activity status. And I love Mike Jaffe for this paper still. And I'm going to show this at the AACR conference on Thursday, and I hope I'm not going to get killed in an instant by all these geneticists who say that cancer is a genetic disease when it is really primarily a signaling disease, i.e. proteins and post-translational modification. So thanks, Mike, for publishing that perspective article. When I read this, I couldn't stop myself laughing for quite a while. Okay, but so what we work in this cancer environment is actually in the area of rare cancers. They are not so rare, in fact. There's about 25% of all cancers are classified as rare cancers, and many of them have no clear treatment. So the physicians just guess most of the time, and it really is almost as bad as this. And that's another big team of people, and we've only started doing clinical proteomics 3 years ago, but it has become a major, major focus in the laboratory, both on the wet side and on the dry side. But what is really exciting about is we have teamed up with an ongoing trial which is called MASTER or INFORM, which recruits, it's a precision oncology program, which recruits patients under the age of 50 and suffering from a rare or otherwise incurable cancer disease. And they've been getting whole genome sequencing and RNA-seq for some time. And 3 years ago, we started putting proteome and phosphoproteome measurements on top of that. A couple of fun facts. We are the one who use least material. We are the one who are the cheapest assay. And we are turning around the data the fastest of all three. So just in case anybody thinks that proteomics can't compete with the genome and transcriptome world, we can. You just have to want to do it and throw some bright minds and good hands onto this. But it really has been an incredible journey. We published 2 papers, wishful thinking. We submitted the 2 papers that describe all this last week. So, and I'm really happy that this is over. But we really, so the idea was we plugged in the phosphoproteomics into an ongoing such clinical system, which is important. You need the physicians on board. We would have never started this without being able to get into an existing program. So we can turn this around in 14 days. For high priority patients, we can do it in 1 week. So what does come out? So by now we've looked at, actually these numbers are outdated. Next week we are going to see the 2000th patient having gone through this system. And the plot on the left-hand side shows we do pan-cancer. So it's agnostic to what type of cancers that we do. So don't expect from this program to get statistically validated results because there are 70 subtypes of cancer in there. Each has 50 or something or less participants. What we're trying to do is place the attention on the individual. That's important. On the right-hand side, just an analytical figure. So the median of number of proteins we get in these patients is about 8,000, and it's about 30,000 phosphorylation sites. But you can appreciate just from the ID numbers, the variation between cancers is huge. It's massive. It's way higher than any of our technical variation. That's a nice thing because we don't have to worry so much about it. Not that we don't try to standardize our experiments to the best extent possible, but the biological variation is so much bigger that it's quite forgiving. And it's interesting to find out that actually the data, when you do proteomics, you can cover already with today's technology, you can cover a lot of the described cancer genes, kinases, or ADs, or antibody-drug conjugate targets. So 2/3 of the things that people might be interested about are actually in the proteomic data. And on the right-hand side is just a UMAP of all these proteomes put together. And perhaps not surprisingly, we can actually, by the proteomic profile, classify the different cancer entities. And from that, we've actually also built an AI model. And so some of these cancers can be predicted from their proteome expression profiles with extremely high accuracy. Not all of them, but some of them can. And this is actually very exciting for a particular type of cancers, which are called cancers of unknown primary. They don't know where it came from, but the proteomic profiles are giveaways where they came from. And that can have an impact on how these patients are treated. Too much on there. The notion here of this slide is when you look at the phosphoproteome in particular, you can tease out the activity of kinases from the measurement. So the idea is that, and we've developed this on 21 receptor tyrosine kinases, they get activated by phosphorylation. then they phosphorylate substrates. And they phosphorylate also themselves. And by looking at the abundance values of either the protein expression or the autophosphorylation that activates them or the phosphorylation downstream of their substrates, we can build simple scores that give us an idea of how active, in this case, EGF receptor is as a kinase. And it turns out that some patients are driven by very high, and we can turn this into a score that is the composite of all these measurements. And some of them are more driven by, or more obvious, the score is driven by the autophosphorylation. Some of it is more apparent on the substrate phosphorylation level. And the interesting thing is actually shown on the bottom right-hand side. Here we look at one patient, the red dot, patient 51, that has high PDGFR and DDR2 topaz scores, but not EGF receptor. And actually this explains why this patient responded to a PDGFR inhibitor called imatinib, but not to afatinib, which is an EGFR inhibitor. And it wasn't clear at the time what this patient should be given, but the treating physician, despite the fact that the genomics people had already said, or transcriptomics people had said, there's overexpression of a couple of these kinases, that physician looked at this plot here in panel E and decided, let's go with imatinib. It's a well-known drug. It's highly well tolerated. And the patient got stable disease for quite a long time. And now the phosphoproteomes actually explain why that happened, because EGFR is not a thing. If you look in panel E on the far right-hand side, that red dot for EGFR is just somewhere in the middle of the patient distribution. There's nothing particular about the activity of EGR in that patient, whereas DDR1 is just on the high side and PDGFR alpha is just on the high side, and both of them are targets of imatinib. So circumstantial evidence suggests that this is the molecular reason why this patient responded. Anecdotal, and we can't do anything about that at the moment. So we have to go through 10,000 patients and follow their treatments over more time before we have any statistics that is strong. Okay, informatics, and I'm now really coming to an end. I hope I'm not overstretching anybody's limit. But this data also needs to be reduced in a massive way so that this can be used in a molecular tumor board meeting, which happens at the end of this 2-week cycle. And 2 members of my team are sitting in that tumor board. And the genomics folks make their case and say, we detected mutations in ABC. We think this could mean ABC. The transcriptomics people state the same. The proteomics people state the same. And a report that we provide is, from all of this data, is literally a PowerPoint slide that looks like this, what you see here. Where's the patient in the distribution of all patients? So was the data quality good? Do we see sufficient numbers of proteins and phosphors? And which of the topaz scorable proteins are high in this particular case? We also look at the expression levels of tumor antigens, of CAR T-cell targets, ADC targets, and all of that information gets put together to discuss in a molecular tumor board what is the most likely cause of driving that cancer, and then together the team makes a recommendation as to what might be the treatment for that patient. That decision lies with the patient and the treating physician. So we only make a recommendation. But the pie chart on the top right is interesting because when we look at and ask the question retrospectively, how often did the proteomic profile agree or disagree with the transcriptomes and genomes? So there's many, many cases it's supporting. So we see the same thing as, let's say, the transcriptomics. And a chunk, the orange part, is where we actually disagree that what people see on the transcriptome doesn't filter through to the proteome at all. And NA is the types of proteins we can't measure. There are still proteins we can't measure. But the dark blue ones is the interesting one, is where the phosphoproteomic data actually made the call. And that's not a small piece. Yeah, so we think this has value. And we hope that the reviewers are going to think the same. And final last work, how I see these things coming together, understanding what these drugs do, and looking at the phosphoproteome profiles of patients is shown here in this heat map now, which is an enrichment analysis of which types of patients show an enriched phosphorylation in which pathways, and the phosphorylation sites that get addressed by the drug names that you see here. So you can do an enrichment analysis from this, and this, in this particular case, these patient group of arms and ERMs, so these are rhabdomyosarcomas, they seem to have high levels of ATR activity, indicating either high replication stress or a good ability to repair DNA. And from that, one can immediately distill a follow-up experiment that says, let's go and test ATR inhibitors in models of rhabdomyosarcoma, because the expectation would be that those patients are likely dependent on activity of ATR, and if we inhibit it, we might be able to kill those cancer cells. You've seen that picture before, so never forget this, and I think still far too few people do chemical proteomics, so I'm also super grateful that this gets recognized by the event today, that this is a worthwhile exercise, and I really, really hope that more people do this. I think the tools are so nicely coming together that this is becoming possible. And again, I've said it now a few times, but I really mean it. Thanks to all of these people who put in brains and hands and time to do all this, and I'm really grateful for this opportunity to speak here and advertise the field, and I'm happy to hear that this is being recognized. And so thanks for that, and I'm happy to take questions if anybody still cares to stick around.
So let's see if we have one or two questions, and we have a lot more time and refreshments and some alcohol that can contribute to the discussion. Let's see, yes.
I have a question about another dimension. You made a very strong, very compelling case for measuring the concentration. So you made a very strong and compelling case for the importance of measuring the concentration and the dose. I'm absolutely convinced in that. I wonder about another dimension, which is time, especially when it comes to measuring phosphoproteome and wanting to understand whether the changes are direct or indirect effects of that, and to what extent do you think it's feasible to combine those two dimensions and to extend into the temporal zone?
Yeah. So the dose-dependent PTM measurements we call decrypt M, like decrypting the modifications. And we do two-dimensional decrypt M experiments, which incorporate time, so that you do dose-dependent measurements along the time axis. Unfortunately, these experiments scale quadratically. That's not a good thing. But in most cases, what we do first is actually a kind of a scouting experiment over time with a arbitrarily high dose just to elicit something, and then pick up from there a relevant time point where we see things happening, and then go in with the dose-dependent experiment there. But to your point, the 2D experiments, actually we've done a whole PhD thesis was expanded on the KRAS system this way, and trying to understand how KRAS inhibitors work or not work, because most of them don't work, at least clinically they don't, and how that is similar or different to the MEK inhibitors and the ERK inhibitors that should all be, according to textbook, RAS, MEK, ERK, and then the rest. It turns out that when you do these 2D decrypt M experiment, you can clearly delineate what is the signaling component and what is the adaptive response. And actually, all of the cell lines that we tested in this way, the decrypt 2D data shows that they are first stopping in the cell cycle, and then they exit the cell cycle to become kind of quiescent, and then they just sit there. And then KRAS can be inhibited as much as you want. It'll not kill the cells. So the time component can be very, very instructive to delineate early from late effects, and then distinguish the actual, say, the direct mechanism of action of the drug, and delineate that from the cellular response that comes later.
Thank you.
So I have Sasha and then Jen.
For the B20 example, you said that your mapping of that downstream network is not going to change clinical practice. But how efficient or is the antibody, the biologic, because they're usually very expensive? Is there not an opportunity there to do best-in-class drugs through small molecule?
Yeah, So I don't know to what extent this has been tried, but all of the drugs actually that we applied made the response weaker. Because when you get a signal down the MAP kinase pathway, and that kills the cell, if you inhibit it, you help the cell survive. So it's actually counterproductive in that sense. So you would have to know quite well how strong you have to inhibit one arm together with the antibody or alternative to that in order to have the desired effect. So I don't know a good answer to that question, actually.
And how often do pharmaceutical companies implement proteomics in drug target discovery? Is it a scary domain that they don't want to know the details?
No. I mean, I think most pharma companies have proteomics in their drug discovery one way or the other. A lot of them farm it out to CROs. Some of them do it in-house. And unfortunately, like in a lot of cases, you don't get to see much of the nice science that happens inside those pharma companies and the methods they apply. And we sometimes get a peek view into this because we do collaborate with pharma.
That was wonderful. I feel bad for asking for anything more, but
sure.
You're sitting with the off targets and all of the things you have. Toxical proteomics just comes and does it ever scream at you? Did thalidomide just say, teratotoxic, or do you see some of that in the data as well?
Yeah, we do. And just by so choosing, we don't work up the tox part, hoping that the classic pharmacologists would take that off our shoulders. We're more interested in turning it around to a good use. But yes, so you run into these tox issues all the time because, as I said, CDK inhibitors are a great example. None of them is selective for any of them, no matter how much medchem people have tried. And then the heralded selective inhibitors for, say, transcriptional regulation, they're actually less selective than the other ones that regulate the cell cycle. And for kinase inhibitors, it's really incredibly hard to make these molecules more selective. But for example, one example case which was quite instructive was BRAF inhibitors. And actually a lot of kinase inhibitors inhibit an enzyme which is called ferrochelatase. It's actually quite an important enzyme. It puts heme into protoporphyrin 9. It puts iron into protoporphyrin 9 to make heme. And there is the genetic disease called erythropoietic protoporphyria. And these people are extremely light sensitive. And that is accumulating of protoporphyrin 9 in their skin. And it really gives them sunburns even without being in the sun. So, and one of those BRAF inhibitors has FECH as an off-target with 1 micromolar KD. And those patients show that phenotype. And when they get switched to another BRAF inhibitor that doesn't have FECH as an off-target, the rash goes away. Yeah, so there's a lot of tox implications in the data, but we have not systematically, at least, gone after them just by choice. But while you're handing over the microphone, Markus Banchev at Cellsome, when he was still at Cellsome, they looked at a whole set of these DILI compounds that are known cases of causing liver toxicity, which is a major problem, of course, of drug failure in their development. And so they apply these kind of measurements to the tox question specifically. And I think the pharma boys and girls should do that.
Hello. Thank you very much for the great presentation. Actually, I have a somewhat controversial question, so please pardon me if it doesn't make any sense to you. So I'm coming more from the data analysis side. I'm a big fan of data reuse. So since you mentioned that phosphoproteome is quite important in rare diseases and cancer, I know that for phosphoproteomics, you have to do phospholipid enrichment, usually in sample prep, but there's also a lot of data that wasn't intended for phosphoproteomics on online repositories for certain rare diseases, cancer, also drug or time dependent. Do you think it might worth the effort to check the samples that were not meant for phosphoproteomics, not enriched, to check for the phospholipid, because I know that you cannot get site-specific information and probably they will be underestimated, but you can still find the somewhat high abundance you should be able to find a somewhat high abundance phosphopeptides. So what do you think about that?
Yeah, so we've asked the question many, many times, how deep is deep enough? Yeah, and that applies to phosphoproteomes. We have the answer now. Go as deep as you can. These full proteomes for phospho in the context of cancer are completely and utterly useless. Don't spend a femtosecond even downloading the data.
Thank you. Yeah, I had some rare disease data. I was wondering if I should check.
You said you have a controversial question. I responded with a perhaps controversial answer, but no. But that is the answer. That is the answer we found by looking across that many cancer patients. It's go deep, and the deeper the better, because the missing data question is nagging at us every day. And that is at the depth of 30,000 phosphorylation sites on average per patient, and we're still not happy.
Well, let's thank Bernhard once again. Thank you so much. Well, thank you very much, everybody, for coming. So we still have some refreshments outside and some drinks. So please don't go away, and I hope that you will be able to talk to Bernhard and to each other about all the interesting things that we just heard about. Thank you so much.